Ladies and gentlemen, welcome to the East Asia Regional Training Workshop on Monitoring and Assessment of Microplastics in Marine Environments. I'm Phoebe Yuefei-Ruan, a Research Assistant Professor from Stakey Laboratory of Marine Pollution, City University of Hong Kong, and also the MC for the morning session. Now, let's invite Professor Kenneth Leung, Ms. Amy Gonzalez, and Dr. James Fang to give us the welcoming and introduction about this workshop. Please.
May I start first? Dear Ms. Amy Gonzalez, Dr. James Fang, colleagues and friends, ladies and gentlemen, on behalf of the Stakey Laboratory of Marine Pollution, PEMSEA Regional Center of Excellence in Marine Pollution, Department of Chemistry of City University of Hong Kong, I would like to offer my warmest welcome to all of you for joining this regional training workshop on monitoring and assessment of microplastic in marine environments. I'm very glad to report that there are over 170 participants who have registered to attend this workshop. As you may know, there are over 8 million tons of plastic being released into our oceans each year. Some of them are broken down into small fragments, becoming microplastics and nanoplastics. It has been estimated that there are about 14 million tons of microplastics in our ocean now, while 1 to 2 million tons of microplastics are discharged from rivers annually in the world. Marine organisms may mistakenly eat these microplastics, causing digestion problems, malnutrition, and even impairment of their reproduction, leading to mortality and population decline. Recent study shows that nanoplastic can even enter the brain of fish, while microplastic can be found in the lung, liver, and kidney in humans. Therefore, in 2017, the United Nations has declared a war against ocean plastic and urged every country, everyone to reduce the use of plastic and hence minimize their impact to marine life and human health. To enable us to manage plastics and microplastic in our oceans, we need to start monitoring them using robust scientific methods while implement policy and measures to reduce plastic pollution. Today, we are very glad to invite 8 promising young scientists who are experts on micro and nanoplastic. Will share their knowledge and skills on the topic, as well as the latest scientific discoveries in the field. As a PEMSEA Regional Center of Excellence in Marine Pollution, our Stakey Laboratory of Marine Pollution will organize more similar training workshops and opportunities for people in Asia on a regular basis. In the future, we will be able to share more on innovative technologies in pollution monitoring and control, eco-safety and risk assessment of chemical pollutants and algotoxins, ecological response to pollution and mitigation measures, ecosystem recovery technology, and scientific diving. You are most welcome to join all these training opportunities in the future. Before I close, I would like to thank our team for organizing this event, especially Dr. James Fang and all the invited speakers. I would also like to thank PEMSEA, Amy, and Karen for their support and co-organization. I wish you all have a fruitful and wonderful workshop today. Thank you for listening. So now, may I invite Amy to say a few words, please.
Okay, thank you. Thank you, Professor Leung. Good morning, everyone. On behalf of Partnerships and Environmental Management for the Seas of East Asia, I would like to welcome you to this monitoring and assessment of microplastics in marine environments. As indicated in the overview that was shared to you by way of a poster, This workshop will provide a summary of the current status of microplastics in the seas of East Asia with some of the figures already provided by Professor Leong. And this is also a collaborative project, and they would like to encourage that you join. We all join is this regional biomonitoring program for microplastic surveillance. And I would like to encourage all of you to join not just this program, but also other programs and training opportunities that SKLMP will also implement in the near future. Now, we in PEMSEA are committed to build capacities of local, national, and regional practitioners of coastal and marine resource management. The idea is that it will help implement a joint regional strategy that was recently approved by country partners called the Sustainable Development Strategy for the Seas of East Asia. Now, this 5-year implementation plan, one of its key programs under the Healthy Ocean component is to tackle waste management and marine pollution, both over and underwater. And there is also a key component under effective governance looking into training and capacity development. Now, to help us with these training and capacity development programs are our learning centers, which are higher education universities and institutions that have institutional capacity and track record to provide academic degree programs and other means of capacity development, research, technical advisory services on sustainable coastal and marine resource development. SKLMP is one of them. SKLMP, as Professor Leong mentioned, is one of our regional centers of excellence. And they have received recognition of high distinction from both their national governments for exemplary performance in capacity development, research, and technical advisory services on sustainable coastal and marine resource development. SKLMP, which is based in Hong Kong, started as MERIT, a consortium which worked closely with PEMSEA on marine pollution reduction and solid waste management since 2008. And today's training actually is relevant and timely because they have designated themselves as SKLMP from MERIT. And because much like climate change and global biodiversity, the marine pollution crisis is a global problem that requires context-driven solutions. Over the last 7 years, we have witnessed many such initiatives in various institutions and countries, but the impacts of these initiatives have yet to be clearly assessed. Have we really moved the needle with all these programs and projects? Now, the concerns on health, safety, and hygiene, particularly during the global pandemic, has seen the proliferation of plastics through, for example, the online delivery of goods and services. There are many reports on this, but we have yet to see data on the underwater, the impacts on nutrient loading and eutrophication, as many of the water quality sampling and analysis, particularly in the region, were either halted or reduced activities. As such, we have a lot of work to do in this field. Now, one of the fortunate outcomes to all this discussion and action and research on marine pollution is that despite the global pandemic, countries have agreed to develop a global treaty on marine plastic pollution. Now, this global treaty requires multifaceted approaches that accounts for the different challenges faced by different parts of the world and adapt to our local context. And this is where training and capacity development is quite important. And this is where I commend SKLMP for being one of the pioneers and have been doing this consistently through the years. We in the region are fortunate to have such institutions who are willing to assist us in applying the research findings and adapt and test it to various contexts. These webinars and learning exchange that have a way of percolating local actions that hopefully trickles back up to the national level to inform policy making. Now, as members of the academe and as young coastal managers, you have the power to do such things through your own active participation in the training, as it will also help the State Key Laboratory, Marine Laboratory, for validating and refining their own research, that then they can also feed into their own global network of scientists which engage in the UN Decade of Ocean Science. So I'm trying to piece all the local, national, and global linkages in doing such a training and capacity development program. So we appreciate the significance in terms of providing local solutions, regional collaboration towards global impact. So with that, I would like to thank SKLMP again for hosting this webinar and all of you for joining us today. And I would like to wish you a productive and engaging workshop. Hopefully next year, we will be able to meet face to face so that, it's better for engaging purposes, because online training is quite limited, still limited, despite the modern advances of technology. So thank you very much. And I look forward to receiving the report of this meeting, and also knowing that you have benefited from such training. And we will continue to be publicizing such initiatives also. And I hope that this training will be recorded, because some actually who are interested are not able to come because, for example, in the Philippines, it's already end of the year. So there's lots of reporting that needs to be done. So there were 2 institutions that actually told me their regrets, but express if you have some recording that we could share it more widely as well. Thank you very much, everyone.
Okay, thank you, Ms. Amy Gonzalez, and she's the Executive Director of PEMSEA. Now, let's welcome Dr. James Fang, the Assistant Professor at Department of Applied Biology and Chemical Technology in the Hong Kong Polytechnic University, who is the chairman of the organizing committee of this workshop. Please.
Good morning. Can you hear me?
Yes.
Yeah. Good morning. Thank you for attending this workshop. Professor Caleb Leung and I have been talking to organize such a workshop on microplastic for a few years. Finally, we have the opportunity here to share our research findings with friends and colleagues. Some of you may be still slightly tired after watching the World Cup final and celebration just a few hours ago. But don't worry, I believe the talks today will be exciting and interesting enough to keep you awake for the whole day. Today, we have organized 2 sections. The first section in the morning will be mostly on the baseline data on microplastic and how microplastic occurring in the urban setting. For example, due to the discard of surgery mask and the occurrence of microplastic in urban water network. After that, we will discuss about potential collaboration in a regional biomonitoring of microplastic in the East Asia region. In the second section in the afternoon, it will be more on lab work. For example, the extraction and quantification of microplastic using some different techniques. Some speaker will even show you a demonstration how we can extract plastic, how we can analyze microplastic from biological sample. In the afternoon section, we will also discuss about the toxicity of microplastic of different particle size at the micro scale and even the nanoscale, their toxicity under different environmental stressor among different taxonomy groups. So we cover quite a large range today. We try to give you a concentrated content today.
So our first speaker is Dr. James Fang. So he is an assistant professor from the Department of Applied Biology and Chemical Technology, the Hong Kong Polytechnic University. So he will present the current status of microplastics in marine environments worldwide. So our chairman, please.
Yeah, thank you. Can you see my share screen, please? Yeah, thank you for coming again. This is the first talk today. I would like to give you an overview of the pollution status of microplastic currently, not only in East Asia, but worldwide. This is a literature review. We have gone through more than 500 papers published in the past few years and extract data for meta-analysis. Sasa Yun, she is my research student. She has done all the hard work, okay? Most of the credit should go to her. Okay. The first slide, how we collect data from. Yeah, we have reviewed published paper on field monitoring of microplastic in aquatic environment in 2016 to 2020, 5 years data. We have categorized this paper into 3 groups. The report of microplastic in water, the report of microplastic in sediment, and in mussels. Why we select mussel here? Because as some of you may already know, mussels are filter feeder. These marine bivalves, they filter feeding a lot of particles, including microplastic, and accumulate this microplastic in their body. Therefore, when we collect mussels and analyze their tissue, it gives you a time integrated level of the pollution of microplastic in the region that you're interested in. From this 3 groups, we have found this number of paper, and from this number of papers, we have extract this number of data entries. Some papers report microplastic data more than one component. For example, some paper report microplastic in both water and sediment. Some other paper report microplastic in both sediment and mussel. Therefore, the number of data entries are larger than the number of paper. We arrange the data like this according to the larger world region. The 2 regions in Asia, which are more relevant to the audience today. Other regions include Oceania, Europe, Africa, North America, South America, and the polar and subpolar region, which include the area in Arctic, Antarctica, and the other nearby closed water system. These are the numbers that we have found, the number of data entries. From here, you can see most of the study focus on water and sediment. The number of paper on marine mussels during this 5 years period is relatively less. This summary is not means to be all-inclusive, but represent the most major paper published in these 5 years. On this slide, you can see where this data entry come from on map. The spatial distribution of data between water and sediment, they are quite similar. Most of the study come from East Asia region, in particular from China. The spatial pattern of microplastic in mussels is still similar to that of water and sediment, but we have a much lower number of paper. If I put a bracket with a number, it tells you the number of data entry in each group. Okay. For example, here water, we have 255 data entry. For sediment, we have 259 data entry. For mussel, we have 33 data entries. Okay. We will use the same style in the whole presentation. Okay. On this slide, I summarize the data in terms of each year. You see there is increasing of number of paper over the years. The blue color here show you the data from Asia, Northeast Asia and East Asia, for example, China, Japan, Korea. The dark orange bar here show you the other region Asia, for example, Southeast Asia and Western Asia. This table gives you an overview of where those data come from. Most of the data come from the two Asia region, as well as Europe. On this slide, we also group the data according to the type of sampling area. We have grouped the data into 5 groups: coastal marine environments, the shallow water environment. The second group is the deep sea environment. The third group is the freshwater environment, which include rivers and lake, freshwater reservoir, and other freshwater system. Another group is the wastewater discharge area or surface runoff, or all sites related to wastewater, including industrial discharge. The last group is only for mussel. This paper got their sample from fish market or supermarket. The dark blue here show you the marine and coastal environment, which play a very important proportion among the 3 groups here. Gray color, freshwater, still quite a lot of paper, in particular in the section of water. This table gives you an overview. Where do we get the data from? Mostly from the shallow water, the coastal marine environment, as well as the freshwater environment. Most of the paper mussel, they were done in a coastal marine environment. Some got from the market, as shown is the light blue color here. Okay, this is how we group the sample. The next slide show you the actual data. Okay, we would like to compare which shape of microplastic is more common than the other. Here we group the shape of microplastic according to the literature definition. We have 5 groups here. Yes, here we have many paper. Each paper, they represent the shape of microplastic. There are a number of shapes. For example, fiber, film fragment, among others. Here in this meta-analysis, we extract the most common, the most dominant shape of each paper. Therefore, each paper, we will have one shape. Here we have many paper, and therefore we have many dominant shapes from many dominant paper, from many paper. Some paper report a range of shape and didn't specify which one is the most common. For those paper, we use black color here. We say there are more than one dominant. Some other paper, they didn't report the shape, which is not reported here. Okay. According to this scheme, we found that fiber contribute to most of the paper. This is the data summary. From this data, we know the shape of fiber and fragment, they are the most important shape of microplastic found in the natural environment, aquatic environment, I mean. The next slide is about the shape of polymer. Okay. This slide is slightly more complicated, but we use the similar logic. In each paper, they report the type of polymer. There will be more than one polymer or plastic usually in each paper. We select the most common one. We select the dominant polymer and use that data for this paper. We have many paper, therefore we have many dominant polymer from all this paper. This is the summary, okay? This table shows you the summary of our findings, the common name and the explanation of what they are. What do we find? We found that PE, polyethylene, polyester, PES, which include PET, and polypropylene, represent three most important polymer types in the microplastic in aquatic environment worldwide. Polyethylene here, the light orange color, which is this bar, you can see it is widely distributed in all groups here. Polyester, mostly PET, the common fiber used for fabric. Which color is it? PES is here, the light blue. The light blue here, you see it is pretty common. Followed by polypropylene PP. PP, the dark blue here in this bar, okay? Still everywhere. Okay, the next slide will be on the method how researcher identify the polymer types of microplastic. On the last slide, we have identified all this polymer. The next slide, this slide here, is to see which analytical method they use to identify those polymer. We have categorized this method mostly in the 4 groups: FTIR, Raman, EDS, and GCMS method, pyrolysis, GCMS. This method can be used alone and combined with other method. Some paper use only one method. Some paper use more than one method. Therefore, you see some combination in the groups here. For example, Raman plus EDS, FTIR plus GCMS, FTIR plus EDS, FTIR plus Raman. Yeah, if the combination is outside this range, or if the paper use other method not specified here, they are grouped into others. Okay, this is the result. Very clearly, the light blue color is the dominant. What is the light blue color? FTIR. Yes, FTIR is the most commonly used method for microplastic in aquatic environment. This is the data summary. Yeah, FTIR almost 60%. Okay. Here I give you 2 more rows here, which tell you the paper, which tell the number of all paper involved the use of FTIR, which is the total of this group FTIR plus this group FTIR plus Raman plus this group FTIR plus EDS plus this group FTIR plus GCMS. Okay, the number here is 65%. The same concept apply to this row. All paper involving Raman spectroscopy is this number, less than 20%. Okay. In summary, in the paper that we have reviewed, more than 60%, close to 70% of all paper, they have relied on FTIR for identification of microplastic. Less than 20% have used Raman spectroscopy. Other methods contribute to a lower contribution. Okay, with this method, with the polymer type identified, the next group, the next data set to summarize is the particle size range. This is tricky. This is tricky because each paper they report, they use different method that have the lowest detection limit of size that they can detect. And some paper, they didn't specify which size range is the most dominant. Therefore, here we use a compromise approach. We group this particle like this, less than a certain less than a certain diameter. And according to the information provided by the paper, we group different size range into our scheme here. In brief, the bluish color, if it is more blue, the particle size less than 1 mm, 1000 micron. If the color is more brownish, the particle size is larger than 1000 micron, 1 meter. The particle size, I mean the major size range. There are other sizes in the data, but if the study report this size range is more common, then we adopt those data. Okay. This is the finding. When you look at the color, I think it is more blue than brown. Yes, it is. Smaller size particles are more common in our data set. The particle smaller than 1 mm is more common than larger size particle. If you look at the data of muscle, it is even more interesting. You see the particle size, the smaller size is increasing abundant in muscle. Okay, it is a clear increasing trend. A particle becomes smaller and smaller, which makes sense. Because if the particle are too large, the muscle cannot ingest it. Okay, because the muscles they can't eat too large particle. Smaller particles tend to be more easily ingested by marine organism or aquatic organism, including mussel. Okay, the next slide, we have a lot of data here. We try to summarize the quantity of microplastic report in this study. This is challenging, but this is what we got. Let's say like this. Each paper, they have reported the number of particle. Each paper report a range of number of particle. In this range, there is the lowest number and the highest number. Each paper, lowest number, highest number. When we have many paper, we have many lowest number and many highest number. Here, we get the median of the lowest number and the median of the highest number. This is the value. Okay, this is median, and therefore there is a range. There's a range behind this median. Okay, and here represent the number of paper that we have extract data from. Some paper, we can't extract data from some paper because it's not available. This number is the number of paper that we try our best to got the data. Therefore, the number here is different from the number from the previous slide. This is what we have got. Here I've highlighted Asia region and South America. It seems that in this region, the number of microplastic in the water body are higher than the other region. In the two Asia region, the number are more reliable because they come from a large sample size, more than 30 paper and 90 paper. For the South American data, we got the data from only 6 paper or 6 data entry. And it is super huge, very high value here. We need more paper, or we analyze data from more years to validate this high level. Okay. How about sediment and mussel? Yeah, this is what we have found. Very interesting. We found that the microplastic in sediment and mussel tend to be high in Africa and North America, and even in the polar region and subpolar region. Again, the number of paper is quite low. I mean, the number of data entries. We need more paper to verify this finding. When you look at the the polar and subpolar region, this number is very large. We all know polar region is remote area, not many human activities there. But they found a super huge number here from this number from 2 paper. So it is the average from 2 paper because the median. So you can imagine this 2 paper, at least one of them, report a even higher number than this number. In that paper published in ES&T, okay, a very good paper. They have concluded the Arctic sediment play an important role as a single microplastic worldwide. Okay, if you're interested, I can send you the link to this page, to that paper. But still, we need more data to validate the finding from here. Similar pattern found in mussels. We don't have any data from the polar and subpolar region. We will go through more years to see if we can find more information about that. It seems to me that the sediment and mussel, they have similar trend in microplastic pollution. Here, very roughly, there's a correlation between 2 data sets, okay? Sediment and mussel. It seems that they are correlated, but again, we need more data to identify if this is true. Okay, what's next? Yes, the smallest particle size. Using the same scheme, the same concept, we have identified the smallest particle size detected in this paper. Okay. Why are we concerned about the smallest particle size? Because even the smallest particle size detected in this paper, it is not that small. It is not that small. I highlight the smallest number. Again, bear in mind that this is median, okay? Behind this median, there's a range. And in many of this paper, the real smallest particle size is 1 micron, okay, 1 micron. But there are more paper larger than this number, and therefore the median come into this number, okay? The smallest median particle size, 50 micron, 75 micron, 21 micron, not that small to me. Because we know smallest particle tend to be more abundant in aquatic environment. So how about the particles more than 20 micron? Do you have enough information to talk about their abundance and their toxicity? Okay, that's all what I want to present. In summary, we have gone through the paper published in these 5 years and yield more than 500 data entry for the meta-analysis. This is only part of the data in an ongoing project that we target for at least 10 years to do a more comprehensive summary of microplastic worldwide. In summary, the Asia region, mainly China, contribute most of the study. Most of this paper collect data from the coastal marine environments. Fiber is the most dominant shape of this microplastic. Polyethylene, the most important polymer among them. Researchers rely on FTIR to identify microplastic, followed by other methods, for example, Raman. The smallest microplastic detector in this paper, in this range, down to 20 micron. However, we know smaller microplastic tend to be more abundant than how about even smaller size microplastic, or even the nanoplastic that will be discussed by the speaker in the afternoon. From this data, we need a more sensitive analytical approach to analyze smaller size particle in a more reliable way. I mean routine monitoring that we can do many samples at the same time. Another finding is the high variation of data. microplastic cannot be directly comparable worldwide. This is important message after the whole literature reviewed. Even we present some data, present some summary, but please bear in mind, this data may not be directly comparable because they have used different method, they have used different particle size range, they are just different. But still, we try to get a summary. Therefore, we need a more standardized sampling approach, which required regional collaboration. That's why we're here to discuss about collaboration today. We propose a regional biomonitoring network for microplastic, which will be introduced by the postdoc in my left, Dr. Marblelock. He will talk about he will suggest a sampling approach for your consideration. My another postdoc, Dr. Derek Ho, he will talk more on the analytical approach, for example, Raman spectroscopy, FTIR, or even the latest OPTIR. He will talk all about this today, okay, among other speakers. Thank you. That's all my presentation.
Okay, thank you, our organizing committee chairman. So now questions and comments are welcome. And also you're welcome to leave the message in the chat box. Okay,
James, may I start a couple of questions? James, will you also look at the correlation between the microplastic number against the human population size in those sampling area? And also, will you be able to link about the plastic amount, the big plastic and the microplastic, if you have those data?
Yeah, for your first question, it is doable to correlate the number of microplastic to human population. I believe the information is available. Yes, available. For the second question about the microplastic and larger size plastic, I'm not sure if the data available, but I will check. Thank you, Professor Leung, for your comment.
I remember I read a paper before. It seems China and Southeast Asia are the major source of plastics. That may be the reason why you see a high level of microplastic in the environmental samples as well.
Yeah, it could be. I haven't gone through a discussion yet. We have just analyzed the data, but we will check more data for discussion.
Okay, good. Let's see if audience have any question. We have 117 people here. Very good number. You can also type your question in the chat box.
Okay, I found one question from the chat box. And Yi says, it seems microplastics in some sediments are much more abundant than that in water.
So there's question you're asking why microplastic in sediment is higher than that in water?
I think so.
Okay. It could be because sediment is the final sink of microplastic. Plastic tend to flow on water, some light density plastic, but eventually most of these plastic will sink to the bottom because of the colonization by microorganisms and other aggregation of other organic matter. They tend to sink at the end. So the sediment should contain a higher amount of microplastic.
Okay, thank you. Then I found another question from Wilma Mocker says, the sediment data here is sediment from aquatic sediment substrate or marine and freshwater. And also wonder for the sediment in polar region, are there any data?
Yeah, the sediment present here, the data come from both freshwater environment and marine environment. Okay. For the polar region, yes, the data are of course available in that paper. We only have 2 papers in the polar region. Yeah, I try to find the paper and send you the link.
Okay, thank you. So I think it's time for our next speaker. So for anyone who have any more message, please leave in the chat box, and we will come back to this question later. Okay, thanks again, James. Then let's welcome our next speaker, Dr. Henry He. Dr. Henry He, he is an assistant professor from School of Energy and Environment, City University of Hong Kong, and he will present a COVID-19 side effects, massive release of microplastics from discharged surgical masks, potentially polluting marine environments. Welcome.
Yeah, thank Phoebe for the introduction. James, I cannot share my screen. Oh yeah, now it's okay. Yeah. Let me see. So, it's frozen. I'm not sure. Okay. So can you see my screen?
Yes.
Okay. Good. So good morning, everyone. My name is Henry. As Phoebe introduced, I'm a member of the Stakey Lab and also an assistant professor at the School of Energy and Environment, CTU. So today I would like to share some personal experience and story about my research on microplastic, which is related to the surgical mask. So it's a quite interesting and interesting story. So before this study, actually, I don't have too much experience in microplastic. It's just some a very it's a coincidence that I stepped my foot into this area. So this is the where I live, actually, the Starfish Bay, Wu Kai Sha. It's in a rural area of Hong Kong. You know, it's you may in your view, Hong Kong may be always the skyscraper, the high buildings, all this stuff. But Hong Kong also have a lot of rural area that with beautiful land and seas. So this is actually the view from my balcony. I love this place very much. During the quiet time, you know, this Bay Area is super calm. So it's almost no one here. But during the weekend, you can see it's all the people, like the families, bring their children and bucket as well, so to flush into the Bay Area for, you know, entertainment, playing water, digging mussels and shells activity, quite a lot. So especially after the MTR, the subway, reached to our area, so the people's more and more people come to this place for the weekend for their entertainment. So it's a very popular beach, and the things actually didn't change too much after the COVID-19 pandemic, because this beach is not a public beach, so it's not regulated by the government. So government will shut down public beach, so no one will be there. But this beach is actually a so-called private beach, and everyone can come. So even during the pandemic, a lot of people playing water there. So I like this beach very much. So I actually jog with my dog every day, the beach. So one day around the middle of 20, I think. So that's one day I jog on the beach with my dog and found the first time I found a facial mask floating in the water, and I see, oh, this might be occasionally someone drop the mask on the beach. However, so I walk further toward the brushes. During the tidal zone, I found more and more. You can see different facial masks. Some of them are N95, some of them are regular surgical masks in the tidal zone, in the rocky, on the brush. So it's everywhere. It occurs to me that these are actually plastics, look like plastic. At that time, I was not sure what kind of plastic for those were made for those masks, but I'm pretty sure they are plastic. So I carried this idea. I came back to my lab and I talked to one of my students, new student. So she just came and she are exploring idea for her thesis, for her PhD study. So I talked to her. I said, now we are in the pandemic and everyone wearing a mask. And I found a lot of masks on the beach today. So probably the mask is related to microplastic, and you may want to look at it and find, try to find it, some idea that you can explore for your PhD study. And she listened to me. So she did a bunch of literature review or exploration, and now it's the beginning of the study. Okay. So some background. At that time, I think this is the updated data I just found this morning. So it's more than 600 million confirmed case and more than 6 million deaths. So it's still ongoing. It's a pandemic, a global pandemic. Hopefully, we are moving out of the pandemic maybe by next year, hopefully. So you can see before the pandemic, actually, the production of the facial mask is quite steady. It's about 5 billion units a year. But during the pandemic, the first year, 2020, it's jumped up to 10 billion production. It's only in China, and most of the production activity were in China at that time. So so these masks literally will be poorly discarded on the beach, in the river, everywhere. And according to one paper estimations that there were 1.56 billion facial masks discarded into the ocean in the first year of pandemic, the 2020, and eventually they will become plastic debris and become microplastics. So that's the only literature we found at that time. So, but very useful. We can use them to estimate the entire discard of microplastic from the surgical mask. So since I'm the first, okay, so the best PPE during the pandemic recommended by WHO is facial mask, like the surgical mask, the ordinary surgical mask, and it's been recommended by the government guidance or enforcement or public self-discipline. So, you know, you can see in Hong Kong and in mainland China. In Hong Kong, it's government guidance. In mainland China, actually, before this month, actually, a lot of places have been lifted for facial mask enforcement, but still a lot of people wearing them. So the monthly consumption in 2020 for the facial mask is, you can see, it's 129 billion. So it's quite a large number. It's huge amounts of plastic being used to manufacture surgical masks. More than 6 of them are surgical masks. Some of them are N95 and even facial masks. By the way, for the fashion mask, you may see all these fabric fashion masks like produced by Louis Vuitton or whatever. So even for fashion masks, they still contains a significant amount of plastics. It's weird, like woven fabric, like woven textile, whatever, but there's a certain amount of plastic to make it rigid in it. So for the surgical mask, 60% roughly, they are mainly using PPs, and very few are PS, so most of them are PP. So if you take a closer look at the surgical mask, you can see there's actually 3 layers for each mask. The outer is spunbone non-woven, like it's usually colored in blue or green or pink, so it's usually colored. So in the middle is the a meltdown non-woven. You can see this is let me remote control. Okay, you can see this is the where is the spotlight? Yeah, this is the middle layer. So it's usually very thick, and it's very loosened. Actually not fixed very well, and easily break down into fibers, which I will mention later on. So the inner is the spunbone non-woven. It's actually the same textile as the outer one, but usually non-colored. So it's give you the impression that the colored should be always out and the white should be always in. So what are microplastics? Since I'm the first detailed presenter, so I will give some background. I'm pretty sure everyone knows it. There's a small particle and fibers, less than 5 millimeters. So usually we know it's some primary microplastic from closed fiber, microbeads, which has already been banned. There are some secondary microplastics from the breakdown of the large pieces of primary microplastic, like a water bottle, fishing net, tire wear, which is very hot research area now. So where do they go? So microplastic degrade very slowly, usually take more than 100 years. They have very high probability of ingestion, incorporation into and accumulation in the body and tissue of many organisms. You may see these pictures like published in 2008 on very famous rainbow runner pictures. So what are the potential harms? There's a lot of research are focusing on the adverse impact of microplastic and nanoplastic. Okay, so they are potentially interfere the behavior of organism, immune system, alteration in gut microbiome. These are very common research area. They can also impair the energy metabolism because the organism, when they take up microplastic, the microplastic will occupy the space in their gut, so they have less room for real food ingestion, so therefore impair their energy metabolism. Endocrine disruption, some microplastic can release plastic additives. So some additives are endocrine disrupting chemicals, and they will enhance the toxicity of micropollutants. So a lot of researchers are working on the adhesive effect of microplastic and other organic contaminants, such as pharmaceutical and personal care products. Also, the synergistic toxicity of microplastic and these chemicals are alarming. So, so in this study, in the following study I'm going to present, we use the copepod as the exposure model. So the copepods mean ore feet. So you can see they have a lot of feet like ore propelling them forward in the water body. So they are a group of small crustaceans, usually very small, like 1 to 2 millimeter. So with a teardrop shaped body and a large antenna. So this is a very nice picture of the Tigris japonicus copepod we use in this study. So they are either planktonic or benzoic, found in nearly every freshwater and seawater body habitat. So why they are very important? So they are important because they are the main food organism for small organisms like Madaca or krill. And they are the most important secondary producer, which called is, so the algae is the primary producer. So they are the secondary producer feeding on algae. So they are the most important secondary produce are even more important than krill, because they are much smaller inside. They are the food of krill, and they are faster growth. So they grow very fast because mainly they distributed in warm water. So the krills are usually distributed in cold water, like the polar area, but they are everywhere. And they grow faster than krill, and they are more evenly distributed all around the globe. So it is estimated that the marine copepods contribute more than all the other groups of organisms together to the global ocean carbon sink. So this is a very important group of organisms. So back to this study, I'm going to present. So the first is asking questions. So when you design a study, it's not limited to microplastic. When you design a study, you need to ask yourself some question, and answer the question by your study, right? So the first question we want to ask about this is whether it's true or false, right? So whether the surgical mask releasing microplastic is true or false. This is the fundamental question we want to ask and answer. So to confirm that surgical mask can release microplastics into the marine environment. This is the first question we want to ask and answers. And second is how big could be the problem. So if okay, surgical mask can release microplastic, but with a very little amount. So what, right? It's not important. So we want to evaluate the magnitude of this problem by the microplastic release by kinetic experiments. In that way, we can calculate how much in total microplastic can be released by surgical mask. We know how big the problem is. And the third question is what could be the consequence? So even the microplastic release from the surgical mask, sure, we got a number. So what again, right? So whether these released microplastic can interfere the organism or not is the final question that we want to answer. So to investigate the potential chronic effect of surgical mask microplastic on the marine copepod as the exposure model. So these are the three questions we ask and answer in the study. So some experimental detail. So these are the clean mask. So the basic model is that we take a different brand of clean mask, and then we submerge them into the artificial seawater in the bottle, and we shake them so to mimic the wave movement of ocean. So we shake them, and then we take them. We take the water, the suspended water, for microplastic to fiber and microfragment identification. We also use FTIR to confirm it's really plastic, and we use a nano measure to count the size and the amount of fiber and fragment release from each mask. And after 9 days shaking, we take the mask out and we examine them under the SEM. We can see some of the fibers have been significantly damaged. So for brand new surgical mask, we took them and shake them and take the water to measure, and we count the release of microplastic every 2 days. You can see this is the kinetic release. So for a brand new surgical mask, they release two different forms, the fibers and the fragment. So for fiber, it's around 79 per day. So it's not a really big number, right? So for fragment, it's more. It's about 210 fragment per day. And this is the size distribution. You can see more than 1/3 are less than 10 micrometer. And around 25% are more than 50 micrometer. So according to these data, we calculate the overall weight lost after 9 days shaking. So the average weight of individual microplastic was 2.8 microgram. So each particles or fragment weight for average 2.8 microgram. And for a single mask, it's usually a minimum 3 gram, maximum 4 gram. Single surgical mask can release 0.88 to 1.17 million microplastic when completely break down. So this is the rough number we calculated from the new surgical mask study. And we also took some discarded surgical masks. You can see these are the example of, we literally picked them up from the beach. You can see we pick up 3 different surgical masks with different degrees of decomposition. So the surgical mask 1 is mildly breakdown, and the number 3 is severely decomposed. So the data is very interesting. So for the fragment, actually the kinetic release curves are very similar for these 3, albeit these 3 looks very, very different, right? So the extent of decomposition is very different, but the fragments release kinetics still remain the same, roughly the same. However, the fiber release are very different, right? The number 3s, this one actually released quite significant, 2,633 per day. So much more compared to the number 1 mask. So as I mentioned, the middle layer is the meltdown layer. So they are actually the text is loosenly cross over each other, and they are very easily being broke down and release microplastic into the environment. So you can see, you can roughly conclude that the more exposure of the middle layer into the environment, more microplastic, particularly the fiber, will be released with a much faster rate. So natural weathering can significantly increase the release of fiber microplastic from surgical masks, but had very little effect on the release of fragments. So the fragments are either on the top or the bottom of the non-woven textile. So now we are going to answer the last question about our study. So what the consequence of these microplastic could cause on organisms. So we used the copepod, the Naupli, which means the baby copepod. So born within 24 hours. So we culture them. You may not be able to see these copepods here, but we culture them in every well of 24 well plates. And until their maturation, we put them male and female mating together, and we take out the fertilized female individually, one fertilized female in one well. In that case, we can actually measure how many offspring the female produce. So once the females confirm fertilized, we will move them into the well, individual well, so we can observe them every day. So this is some example picture for your reference. So this is the Naupli, so the baby stage. This is the male. You can see the male usually with a very large antenna, and females are much small. You can see, and actually they look much more distinguished under the microscope. You can easily distinguish a male from a female. So the exposure solution is 1, 10, and 100 microplastic per milliliters, and the condition is 21 degree with 30 PSU and 12 light dark cycle. So we expose them and count the offsprings as well as the developmental stage. Okay, so once the Naupli being put in the microplastic contaminated water, we will observe their development. So to count how many days they matures, and once we put the fertilized female in the water, we will count how many days they do one delivering. So which means once they produce a brood of offspring, waiting for another several days. So we will count the number of the waiting days and for the second brood, as well as the number of offspring. Okay. So these are some of the data. So this is the effect of the microplastic on the development of coffee pot. You can see usually they will for the Naupley stage, so the negative control will take about 4 days. But if you put them into 100 microplastic per milliliter, it will take a little bit longer, but these data are not significant here. But for the maturation, you can see significantly difference. So around control, it take about 10 days. But around the exposure, it will take significantly higher, more than 10 days. So for Naupley stage, it's time from the birth to the copy pot stage. But the sexual maturation stage is the time from the birth to the time it's producing the first ovispec times. So these are the statistical analysis. You can see we can conclude that exposure to microplastic, the surgical mask microplastic, can delay the maturation of coffee pot. And for this is the reproductions. So it's very similar. I see the fecundity means the average number of spring per brood. Every time how many offspring the female produce. The birthing, the birth spacing is the average interval between the two reproduction. So for short, generally, exposure to surgical mask microplastic decrease the average number of spring per boost and elongate the birth spacing. So they produce less offspring and take more time to produce the next bruise. So the effect of surgical mask microplastic on the excrement, the feces, the excrement lens of coffee pot, you can see exposure to the microplastic result in longer excrement. Okay, this is the fluorescence pictures that we dye the microplastic using Nile red. So you can see for the blank, it's very clean inside the body of the coffee pot, but for the microplastic exposure group, you can see there's a lot of microplastic stuck in the gut of the bug. So as a result, these microplastic actually occupy the space in the gut of coffee pot. It does impair their ingestion, nutrition, and energy metabolism. So there are some regression analysis we took. So regression analysis show that exposure to microplastic has decreased the fecundity and increased the birth spacing and increased the length of excrement. All three are very significant. So as a result, the developmental retardant and the decreased fecundity could result from the insufficient energy uptaken by the coffee pot due to the ingestion of microplastic instead of the real food algae. So some highlight of our study, it is estimated that the surgical mask discarded throughout the year of 2020 could ultimately lead to 1,370 trillion microplastic entering the coastal marine environment globally, with a release rate of 396 billion microplastic per day. So these are a huge number, right? The numbers are so huge that we might not be able to vividly reflect its severeness. So so it's very common for hello, sorry. I will conclude very soon. I will conclude it very soon. Okay. So as an analytical chemist, we usually use the swimming pool as a quantified number. So this is equivalent to seriously polluting 54,800 Olympic swimming pools of seawaters. And the result also revealed that the copepod ingested the microplastic, causing a significant decline in fecundity up to 22%. So it's a quite significant number. The surgical mask, microplastic release from poorly discarded mask have the risk to exert a long-term domino effect on the ecosystem. So once the copepod out, there will be nothing to control the algal blooming, and there will be in short of food for fish. So to minimize the risk of this emergent threat, better environmental management policy and the law enforcement for ensuring the proper disposal of surgical masks are indeed necessary. So this paper has been reported by a bunch of media and has also been published on EST letters with the front covers. So if you're interested in the detail of this study, you are welcome to email me. That's the end of my presentation. I would like to thank the Stakey Lab and the SEE for the support of this study. Thank you very much.
Okay, thank you, Henry, for the great presentation. Now, a short question or comment is welcome. So from the audience, please. And now, may I read one comment from the chat box quite early? From Bella Despasseri, sorry for my pronunciation. So here is a question about research microplastic can stimulation in human body. So what the effect, and is there already accredited research about that? So something about microplastic stimulation with the humans?
Oh, I think I'm not a real expert in that area. Maybe James Fang can answer that question better. But I will share some a perspective of my personal experience. Actually, for a very similar study of this microplastic release, there was another study on I think on the Journal of Hazardous Materials. They actually quantify the number of microplastic potentially inhaled by human body through the mask. It's a very interesting paper. So when we take the mask, wear the mask, actually during the breathing every time, it's potentially we will breathing microplastic deep into our lung. And if that microplastics or nanoplastic stuck in the lung, it will cause long-term effect. Right now, there are, I don't think there's any human model study, but there are some animal study and in vitro study showing that microplastic may cause oxidative stress because, you know, the nano size of the plastic embedded on the cell membrane, and they will cause them result in the generation of reactive oxygen species. That's possible. Another possible angle is the release of endocrine disruptive chemical from the plastic. You know, all the plastic will use those additives. So these chemicals further release from the microplastic may cause some additional toxicity. Yeah, that's the two things I can think about now. Maybe James, you may have some additional thought to put.
Yeah, sure. Thank you, Dr. He. Yes, so for human effect of microplastic, the information currently is still quite limited. We need more study to evaluate the effect. But the presence of microplastic in the human body has been confirmed by different researchers worldwide. For example, in our lab, we have found microplastic in the human gut and in a human fecal sample, which is evident to show the importance of this topic, why we want to study the effect of microplastic. Yeah, that's my response.
Okay, thank you, Henry and James. I also noticed there were 2 more messages in the chat box, but since the limit of time, so Henry, would you please reply to them in the chat box also?
Okay, sure.
And now comes to our next speaker, Dr. Kai Zhang. Dr. Zhang, are you here?
Hi, yes, I'm here.
Yes,
I'm going
and
to
Dr.
share
Kai
the
Zhang
screen.
is an assistant professor from Department of Environmental Science and Engineering, Macau University of Science and Technology. And he will present microplastic occurrence in urban water networks of coastal metropolis and the implications for marine microplastic pollution. So Dr. Zhang, please.
Okay. Can you see the screen?
Yes.
Okay, thank you. So good morning, everyone. Today, my topic is the microplastic occurrence in urban water networks of a coastal metropolis and the implications for marine microplastic pollution. And I'm Kai Zhang, and thanks for Phoebe's introduction of me. So I'm going to introduce the study in the following 5 aspects. Now, first, the introduction. As shown in this picture, we can see that there are many pathways for introducing microplastics into the ocean. And the sewage treatment works are believed to be one of the biggest sources of microplastics. The stormwater is separated from the sewers and untreated before discharged into the ocean in Hong Kong. So the stormwater drains could be another pathways for microplastic transport in coastal cities like Hong Kong. But little information is known for this kind of pathways. So if we know the sewage treatment work is the biggest sources for microplastic transport, and how about the stormwater discharge? In many cities, the stormwater were combined with the sewer and treated in the sewage treatment works, and then were discharged into the environment. But in Hong Kong, they are separated. So we would like to evaluate the microplastic load in these 2 kind of pathways, the sewage treatment works and the stormwater discharge. And before we could evaluate this value, we need to know the sewage treatment works, the removal efficiency of different treatment levels. So based on this background, we could come to our objectives. The first one is to investigate the fate and the abundance of microplastics at different stages of the sewage treatment process and determine the removal efficiency of different processes in the sewage treatment works of Hong Kong. The second objective is to analyze the seasonal variation and trend of microplastic pollution in the sewage and the stormwater samples through a more comprehensive monitoring program. The third objective of this study is to fill the knowledge gap on the sources and emission of microplastics discharging into the ocean via local sewerage and stormwater drainage networks. And the fourth objective is to evaluate the microplastic loads and compare the current results with other studies worldwide. The methodology part consists of 4 aspects: the sampling location and sampling method, pretreatment method, and the classification and identification method. The sampling location for the sewage treatment works. The 3 sewage treatment works were selected for this study. The Stonecutters Island is a chemically enhanced primary treatment works in Hong Kong, and 4 sewage sampling points were selected in this sewage treatment works. And Sha Tin sewage treatment work is a secondary sewage treatment work in Hong Kong, and the She Wu Hui sewage treatment work is a secondary and tertiary sewage treatment work in Hong Kong. So the survey consists of 6 rounds of sampling, 3 rounds in dry season and 3 rounds in wet season. For the stormwater samples, 6 sampling sites were selected, which is our Fo-Tam Nala, Mutong River, Tim Shui Wai Nala, Chunmen River, and 2 box culverts at Shaoqi 1 and Kuaitong. Same with the sewage treatment works survey. This survey also consists of 6 rounds of sampling, 3 rounds in dry season and 3 rounds in wet season. The criteria to select the sampling sites are described in this slide. For the sewage treatment works, the 3 sewage treatment works represent the 3 major types of treatment levels of sewage treatment works in Hong Kong, which are chemically enhanced primary treatment and secondary treatment and tertiary treatment, respectively. And also, these 3 selected sewage treatment works have highest sewage treatment capacity among the sewage treatment works in Hong Kong. And for the stormwater discharge sampling point, these 6 sampling points covers Hong Kong's major large catchment, different levels of urbanization and land uses. And most importantly, we should consider the interference of the tidal current. So mostly these 6 sites were selected by the Drainage Service Department because they have the data of the catchment area and the current interference data. For the sampling method, as we know that there is no standardized sampling method for the microplastic collection in sewage as well as in stormwater discharge. So we took the literature review to show you some of the studies, the commonly used sampling method. From the right side sheet, we can see that in different studies, the mesh size are different, the sampling as well as the sampling volume. And for the stormwater discharge, there is not many studies to make reference to. So we use the seawater sample, seawater studies to make the reference. The study list here, the commonly used sampling device is a bucket and the pump. The sampling volume ranged from 5 liter to 3,000 liter, and the minimum size of the microplastics ranged from 10 micrometer to 63 micrometer. So based on this data, and also based on our pre-test, here comes to our sampling method. So we use the stainless steel bucket to collect the sewage sample. And for the different size of the microplastic samples, we use a different collection method because of the suspended particle content in the sewage samples are very high. So for microplastics larger than 300 micrometer, we use the bucket to collect the sample. The sample volume range from 60 liter to 400 liter based on a different treatment level. And we use the stainless steel sheath to sieve the sample on site. And we collect the sieved sample in a glass bottle. And for the microplastic smaller than 300 micrometers, we just directly collect the sewage sample in a glass bottle. And we take the subsample to analyze. And for the sludge sample, we use a stainless steel shovel to collect the dewatered sludge and wrapped in our aluminum foil and sealed in a Ziploc bag. For the sewage sample, the sample were collected 4 times with 3-minute time interval, and multiple grabs of sample were considered as a single sample for analysis. We collect sample like this because we want to collect representative samples for the sewage, for the sewage. And for the stormwater samples, we still use the stainless steel bucket, but for the microplastics smaller than 300 micrometer, we did not take the subsample. Just all the sample were sieved through the plankton net. And after the sieving on site, the particles remained on the sieves were transferred to the glass bottle and then transported to the laboratory for later analysis. So for the microplastic sample all larger than 300 micrometers, we use the sieve to sieve the sample again and use the hydrogen peroxide to digest the organic matters and use the separating funnel to conduct the density separation. And then the supernatant were filtered onto a filter membrane for later analysis. And for the microplastics smaller than 300 micrometers, we directly digest with the hydrogen peroxide and directly filtrate it onto a filter membrane. And for the sludge samples, firstly, we will freeze dry the sample and conduct the sieve again and use the digestion, use the hydrogen peroxide to digest the organic matters, and also you conduct the density separation. And for the suspected microplastics, we, according to their size, shape, and color, we could categorize them into different groups. And the chemical composition of the microplastics were confirmed by the FTIR and micro FTIR. So for the particle larger than 300 micrometers, we use the FTIR ATR. And for the particles smaller than 300 micrometers, we use the micro FTIR to identify their chemical composition. Now let's have a look at the results. The figure shows the abundance of microplastics in different sampling sites of the sewage treatment works. As we can see that the abundance fluctuated in many sites, so we could not have a clearly pattern from this figure, but still we can see that less microplastics in higher treatment level can be found in this figure. So we do average to show the dry season and wet season pattern. From this figure, we can see that the microplastic abundance are higher in dry season than in the wet season. And we can see that the highest microplastic can be found in the influence of the Sherwood sewage treatment work. So based on the equation of the removal efficiency, we can calculate the removal efficiency of all the investigated 3 sewage treatment works. We can see that the removal efficiency are all over 80%, which means that over 80% of microplastics can be removed by these sewage treatment works. And the secondary treatment could remove most of the microplastics in our study. And the tertiary treatment, which is the membrane bioreactor, could have a higher removal efficiency, which can be as high as 98%. And we did a count balance analysis. The difference can be calculated by the number of microplastics in the sludge minus the microplastics removed in the sewage treatment process. And the positive number denotes that more microplastic retained in the sludge than removed in the sewage. So the total average difference in microplastic numbers are all positive, which means that there are more microplastics remained in the sludge than removed during the sewage treatment works. During the sewage treatment, there are 2 possible reasons for this phenomenon. The first possible reason is that the sludge treatment could facilitate the breakdown and the fragmentation of microplastics in the sludge. And the second possible reason is that the longer retention time of the sludge could also facilitate the higher accumulation of microplastics in the sludge. Now let's have a look at the results of the stormwater samples. We can see that the site number 3 have the highest microplastic abundance, no matter in dry season and wet season. Now this picture shows the site number 3. There are many garbage along the river bank, and also the low flow velocity of this site could facilitate the accumulation of microplastics in this nala. Now we also take the average to show their seasonal variation. We can see that also in dry season, the dry season have a higher mean value of microplastic abundance than in the wet season. So according to the characterization of the microplastic collected in this sample, we could conclude that smaller sized microplastics were dominated in most sewage water sample, but only dominated in site 2, 3, 5 of the stormwater sample. And the proportion of larger microplastics in stormwater sample increased in the wet season, indicating that more microplastic of larger size microplastic could be transported into the stormwater drains in wet season. And for the chemical composition, we can see that the PET and the PP were the most abundant plastic type in most sewage sample, and the PE, PEP, PP, and the PEPP copolymer were the most abundant plastic type in most stormwater sample. For the shape categories, we can see that the line and fiber was the predominant shape in most sewage samples. Also, the line fiber was the predominant shape in site 4, 5, 6 of the stormwater sample, but the fragment together with sheet gives a higher proportion than fiber in site 1, 2, 3 of the stormwater sample. And for the color, the transparent was the dominant color in most sewage samples. And if we combine the white and transparent colors, they could be the dominant colors of microplastics in most stormwater samples. As we know that there are far more than 3 sewage treatment works in Hong Kong. So if we use the microplastic data from the investigated facilities to extrapolate to those facilities without microplastic data, we can estimate the total microplastic discharge from the sewage effluent. And here, on the right column of the table, we can see that if we use the data of the sewage treatment capacity of the 12 sewage treatment works, we can have over 20 billion particles per day will be discharged into the ocean from the sewage treatment works in Hong Kong. But for the stormwater drains, as we do not have the flow velocity data of all the stormwater drains, so we could not extrapolate to other stormwater drains. Now we use only the 6 sampling site to evaluate, to estimate the total microplastic discharges. We can have the left column of the data. So although in previous slide I mentioned that the mean abundance in stormwater drains was lower in wet season than in dry season, but from this column we can see that the estimated discharge was higher in wet season than in dry season. So by comparing the microplastic discharge, we can conclude that the stormwater drains could be another important pathways of microplastics within the coastal marine environment. Now, the fourth part is to compare the our data to the data worldwide. Here is the influent data. We compared with 61 global data with the average of over 1,000 particles per liter. From this average, we can see that the three data in our study were all below the global average. But as we know that the different size were reported in different studies, and with the smaller size reported, there are more abundance of microplastic can be found. So we focus on the smallest reporting size of 20 to 25 micrometer. We can have a right-hand side picture. Still, in our study, the data is well below the average and the median number of the other studies. Same to the effluent data, we also compared with 75 global data. We also have a same conclusion, is that our data were well below the global average. And for the sludge samples, there are only 12 data available at the time. So we can see that the Stone Cutlass Island below the global average, but the Shating and Shewuhui sewage treatment works were above the global average. This may imply a better microplastic removal in the local sewage treatment work. Now let's compare the removal efficiency to the global studies. As we can see that our primary sewage treatment work have 80%, over 80% of the removal efficiency, and that is higher than the global average. The secondary sewage treatment work in Shating and Shewuhui, it's almost the same to the global average. The tertiary sewage treatment work in Hong Kong also have a higher removal efficiency than the global average. It's not easy to compare the stormwater drains of the world, because at that time, there's not much studies focusing on the stormwater drains. But if the stormwater drains could be categorized into the urban drains and the natural rivers. So we have searched the study that so our sample stormwater channels shall not be directly compared with the natural rivers due to their difference in nature, but could be categorized and compared with urban drains. So we searched the study focused on the urban drains and have the comparison. Now here is our result. We have compared with 10 global data of urban drains. The microplastics abundance in all sampled locations are below the global average and the median. So here comes to my conclusion, is that the microplastics were all detected in the samples in sewage treatment works and the stormwater drains. The sewage treatment work, for the sewage treatment works, higher treatment level could remove more microplastics in the sewage. According to the count balance analysis, the microplastics, the number of microplastics in sludge are higher than that removed during the sewage treatment. And the removal efficiency of microplastics for primary, secondary, and tertiary performed at or better than the global average. And for the stormwater discharge, the proportion of large microplastics in stormwater samples increased in the wet season, indicating that more microplastics of large size could be transported into the stormwater drains in wet season. The microplastic abundance in all 6 stormwater drains are lower than the global average. So again, I would like to emphasize that the sewage treatment works and the stormwater drains are both important pathways of microplastics within the coastal marine environment. So more study focusing on stormwater drains should be conducted in the future. So this study have been published in Water Research. So if you are interested, please, more details can be found in this paper. So thank you. I'm happy to answer some questions.
Okay, thank you, Dr. Zhang. Nice talk. Now questions and comments are welcome. And I noticed one message from the chat box that Ronia Shem would like to ask how significant is the number of particles discharged per day if we consider from an ecology human health perspective.
I would like to see that.
Oh, okay.
Okay.
There are two questions.
I think the question is not very easy to answer, so maybe I could answer the question later and by typing. Is that okay?
Yes, sure.
Yeah.
And there were another one from her for about the removal efficiency. Can you see it?
Oh, I can see the second question. Actually, I think we still cannot, you know, when we isolated the microplastics in the laboratory, we could not know how the product to generate these microplastics, which means we don't know whether they are the white webs or the microplastic containing personal care products. So we just got the microplastic in a sample. We don't know where they come from. So the question, yes, I could be thinking that this might be by flushing these microplastic containing product into the residential drains. It could be possible reason for the higher microplastic abundance in the sewage, but we don't sure whether the real sources for these microplastics.
Okay, thank you.
Okay.
So for other audience, and you may also leave your message in the chat box.
I will see the chat box and answer your questions by typing. Thank you.
Okay, thank you, because the limited time. So let's move on. And our next speaker is Dr. Alisodi Mabolok. He is a postdoc from Department of Applied Biology and Chemical Technology, the Hong Kong Polytechnic University. And he will present introduction to the regional biomonitoring network for microplastic surveillance. So because he has some urgent issue to handle, and his colleague, Dr. Derek Ho, will play the video for him about this presentation. So please, Dr. Ho.
Okay, thank you. Now I'll play his recording of this presentation. So.
Yeah, hi everyone. Let me explain a bit. Dr. Mabolok, he had just returned to the Philippines yesterday, and he said the weather condition in his city is very bad, and connection is very bad too. He tried to connect to the online meeting, but he couldn't. That's why he asked Dr. Derek Ho to play the video for him. He is very well prepared. He could foresee the situation like this, that he prepared the video beforehand. I appreciate.
Okay, let me play now. Yeah.
Good morning. I'm Elizaldi Mabolok, and I'm here today to talk about the introduction to the Regional Biomonitoring Network for Microplastic Surveillance. So this is the presentation outline of my talk. First is the general introduction on plastic pollution in Asia. This will be followed by the establishment of the Regional Biomonitoring Network, on which I will briefly discuss the objectives, methods, as well as our regional sites and collaborators. And lastly, the sharing of the preliminary results in mussels and oysters collected in Hong Kong and in East Asia. So to start with, plastics are known to be low cost. They are durable and lightweight, and in fact, they have a wide range of applications from domestic to industrial. There is a growing demand, which actually resulted to the increasing production of plastic. As you can see in this figure, the y-axis is the production volume in million metric tons, and the x-axis is the years. And in 2021, global plastic production reached around 390 metric tons. Unfortunately, the increasing usage of plastics also resulted to the increase in plastic pollution in the environment. And based on the annual plastic waste, the United States is taking the top spot, followed by India, China, Brazil, Indonesia, and so on. At present, around 400 million tons of plastic waste are being produced every year. And this figure actually exceeded the primary production since it can incorporate the production from the previous years. However, most of the world's marine litter and plastic pollution is actually around Asia. And this could be due to the gaps and limitation of waste management programs in the region. And these marine litters and plastics that are being introduced in the water or in the marine environment actually can be broken down into smaller particles via photooxidation, chemical and mechanical degradation. So these microplastics, or as what we call them, can measure less than 5 millimeters, and it comes in different shapes. We have pellets, fragments, fibers, and foams. And in recent years, microplastics are getting a lot of attention because of the potential risk that it can bring to the marine organism and in the environment. For example, these particles actually can be easily ingested and bioaccumulated, and it can be transferred from one organism to another higher trophic animals along the food chain. And in some organisms that were tested, microplastic actually can disrupt the physiological processes of the organisms. So your seafood, such as bivalve shellfish, like your oysters and mussels, are actually example of the groups that have high risk to microplastic contamination because of their filter feeding nature. So these oysters and mussels actually are being cultured and harvested for human consumption. Oysters and mussels, for example, which is very popular seafood in Hong Kong and Asia. And take note that in the same region also, it is known to be one of the marine hotspots for plastic pollution. And at the same time, there's a heavy seafood consumption among its population in the area. So it is therefore necessary for us to increase our understanding in terms of sources, characters, and impacts of this plastic in the region, and in order also for us to develop good mitigation measures to address the problem. So in this connection, the regional biomonitoring network for microplastic surveillance was conceptualized. It started in 2019, headed by Assistant Professor James Carvey Fang of the Hong Kong Polytechnic University. So in case there are interested audience who want to be part of this network, please do not hesitate to contact Dr. James Fang. This is his email address, so you can send him an email anytime inquiring about the program. So to go back about the project, so the objectives of this network is very simple and straightforward. One is to determine the microplastics in edible oysters, such as your Magallana species, and mussels, especially on Perna viridis, and to provide the first region-wide baseline database. In order to standardize the method, so this is the monitoring network sampling plan on which we will collect oysters and mussels at the same site or sites in case the collaborators have multiple sites. Once every year, preferably in the summer. So at least 30 oysters and mussels with similar sizes per species per site will be collected, and we will record all the important details from sampling date, measurements of the lengths and widths, including the measurements of the tissue fresh weight and tissue dry weight. So the collected oysters and mussels will be then analyzed for microplastic contamination. I will not talk about the microplastic extraction and quantification because Dr. Yun-Hwa Ho, he will talk this in detail. In fact, this afternoon, he will present extraction of microplastics from marine biological samples under polymer identification using Raman spectroscopy. So I hope you will not miss his talk because he will really focus on the preparation and extraction, including all the methods for identification of microplastics. So these are our regional sites. So we started in 2019 and we able to establish sampling sites, of course, in Hong Kong and in Taiwan. On this time as well, we have collaborators that joined the program from mainland China. And in 2022, we have collaborators and established sampling sites in Japan and in South Korea. And in 2021, we have new collaborators from Vietnam, Thailand, Malaysia, and Indonesia. Right now, we are still in the process of finalizing our collaborators and sampling sites in the Philippines and in Papua New Guinea. And just recently, we have new team that will do the collection in Australia and in New Zealand. So to give you some of the preliminary results, I'm going to talk about first the microplastics in the edible green leaf mussel Perniviridis. So in this presentation, so the mussels were mainly collected in eastern part of Hong Kong. So we have 5 different sites and we collected 10 mussels per site. So the oysters were digested using 10% potassium hydroxide, 30% hydrogen peroxide, and 14% EDTA, or what we collectively call as KHE. And then the microplastic identification and quantification was done using the automated mapping technique of Raman microspectroscopy. So for the results, so in this figure, you can see the y-axis is the sampling sites and the x-axis, the microplastic concentration. So in both figures, you can see that the mussels from Tulu Harbor, so this is the location of the Tulu Harbor in the map. The mussels from the Tulu Harbor contain the highest microplastics in terms of number per gram weight and per individual. The most common type of polymer that was identified is polypropylene. We also identified polyethylene, polystyrene, and PET. In terms of shapes, fragment is the most common with some fibers. And if we're going to sort out for polymer type, fragment is still the most common, followed by fiber. Fragment is the most common and followed by fiber. So if you want to have a more detailed picture of the study, it is actually already published. Just look into Leong et al. 2021 to have in-depth description of the study. Now for the oysters, so with the help of our collaborators, we collected oysters in the different sites in Chinese region from Taiwan and in mainland China. So we also followed the same protocol in the extraction and determination of microplastic. So the result, so in this figure, you can see the y-axis is the number of microplastics per individual, and the x-axis is the sampling sites. So the oysters from Ningbo and from Keelung in northern Taiwan had the highest microplastic abundance per individual. If we're going to measure microplastic per gram weight, the oysters from Keelung had the highest abundance of microplastic. We also measured the oyster condition index of the samples, and we found out that the oysters from the same site in Keelung had the lowest oyster condition index. However, we did not find a significant correlation of the microplastic concentration in terms of oyster condition index, weight, and shell index. The most common polymer type that was identified includes the polystyrene. You have your polypropylene and PET, and we have also polyethylene. And in terms of shapes, fragments is the most common with fibers. In terms of microplastic size distribution, we found out that microplastic around less than 500 microns is the most common, or the highest percentage, with some microplastics that quite longer, above 100 to 150 microns. And if you look into the microplastic size distribution across sampling sites, we found out that microplastic size is very variable in all the sampling sites. So for the summary, we found out that highest microplastic concentration were found in Tulu Harbor in terms of the mussels, and highest in Ningbo, in Xingshan Harbor, and Keelung City in Taiwan for the oysters. And this is due to its greater proximity to human settlements and activities, given that this area that were identified are in close proximity to human settlements. The most common type of polymer that were identified includes polypropylene, polystyrene, polyethylene, and polyethylene tetraphthalate. So these polymers are actually commonly used in food packages, such as your single-use cutlery, carrier bags, and among others. And this polymer actually mostly in fragments and in fibers. So that's the end of my introduction to the Regional Biomonitoring Network. So again, if anyone in the audience that is interested to be part of this network, please contact the project leader or the PI, Dr. James Curry Fang, and this is his email address. So you can send in an email and you can inquire and ask in details about the program. So that's for my presentation. And if you have questions, do not hesitate to ask at the end of the session for the morning. Thank you.
Okay, thank you, Dr. Marbolok and Dr. Ho. And audience, please, you are welcome to leave any message in the chat box for questions and comments. Okay, so if no, then let's move to the next section. So it's now it's time to have a discussion with the morning speakers on the monitoring approach for microplastics in marine environments. And let's invite Dr. James Fang as the moderator. So Dr. Fang, please.
Yeah, hello everyone, this is James again. This session is, it can be a Q&A section. If you have any question asking any speaker in the morning, this is time for you to ask. If you don't mind, you can turn on the microphone and we can talk more directly. But if you prefer sending us a message, it is okay too. It is okay too. Yeah, so please, if any of you have any question, now's the time.
And if there are also any unsolved questions in the chat box, so please raise your hand or leave your message again, because I may skip some of them unintentionally.
Yeah, sure. For all the speakers this morning, if you want to answer any question to the audience this morning, now is the time for you to answer the question. Perhaps let me start first. I've seen a message by Catherine asking about if it is possible for undergraduate students to conduct microplastic assessment using basic equipment such as compound microscope. I think so. We can. Let me tell you my experience. In the last 2 years, my lab has been working with WWF Hong Kong we carry out a microplastic workshop for secondary school students in Hong Kong. We have been working with more than 10 secondary schools. We designed some basic protocol for students to observe microplastic under a microscope. And we also introduced to the student how we do the assessment of microplastic. In that particular program, we have arranged field trip for students. We go to the sampling site with the secondary school student. We do the sampling together to give them hands-on experience. All these activities can be organized with a very low budget. It is okay for them to learn. They are very keen, they are very exciting. And when we define microplastic can be quite large. The definition is up to 5 mm. So for secondary school student or even primary student, we may use the plastic size larger than 1 mm that you can measure by ruler. Okay, for example, we can design some teaching materials for kids and students. That's my experience. I know there's a representative of WWF here too, and I've seen her asking questions. So if WWF representative want to add on anything, please feel free to discuss with us and share your experience. Anytime, no pressure. Okay. Now, other speaker, do you want to reply to do you want to answer any question by the audience? Yeah, I see someone raising hand. Yes, please feel free. Feel free to ask.
Hi, good afternoon. Good
Good
afternoon.
afternoon.
I am Rodolfo from Mindanao State University in Iligan Institute of Technology in the Philippines. We're currently working on atmospheric microplastics, and I've encountered this training, and thank you for organizing this one. But I have a question because it seems that microplastic studies are not well standardized in terms of protocols. Is there a push in Hong Kong academics or PEMSEA to standardize this the procedures?
Yeah, let me answer this question first. This is a very good question. Currently, we don't have a standardized method or standardized protocol to analyze microplastic. Even for the sampling, we don't have a standardized protocol, which is very important because if we use different protocol, the data cannot be directly compared, as indicated in my presentation this morning. In my group, we work on aquatic environments, but then have other colleagues working on microplastic in the atmosphere, in the air, which is your area, I think. Yeah, it is possible. We can work together to set a standard protocol that will be used by other researchers too. Setting a standardized protocol is actually the objective of the Regional Biomonitoring Network Program presented by Dr. Marbolog just before this discussion session. He and I are trying to propose a standardized protocol for collaborators in the East Asia region, including Australia and New Zealand, getting microplastic from aquatic environments and standardize everything and get the data published. Yeah, I believe the same concept can be applied to microplastic in the atmosphere and in other environment. That's my response.
Thank you, Dr. Fang.
Perhaps let me ask some more questions. Dr. Henry He.
Oh, yes.
Yeah, your talk is very interesting. May I ask your comment on any suggestion to reduce the release of microplastic from the surgery mask?
Well, first of all, I think the government should first to stand out to pay more attention about the discard, the unpurposed discard of surgical masks. I see a lot of during the pandemic, a lot of people still running and jogging outdoors, and when they run and jog by legal, they are allowed it not to wear a mask. So sometimes they just strap their mask on their ears or their arms, and it's very easy to be accidentally released. So once so for those joggers, once they drop a mask, they can take a new one and put them on when they finish the exercising, but the mask will be left on the beach. So that's the problem. So I think we should the government should issue a regulation or a guideline that teach people how to properly temporarily store their masks, for example, in an envelope or a zip bag that they can carry out in their pocket or handbag, for example, and like in other environments, the masks should be properly discarded. If the masks are going to be discarded, they should be properly discarded into the garbage bin. With lid, for example. So in that case, the wind will not blow because the mask is very light. So if certain wind can easily blow those masks into the ambient environment. And for other cases, I think the mask is made of plastic. I think we can also consider a recycle of those material, because the masks are they are made from the petroleum products. It's actually very high quality plastics. So the waste is also the resources from a different angle, right? So maybe we should consider recycling the mask. Of course, there is a hygiene issue, because the mask may contain virus and pathogens inside. But at least in the like some restricted area, for example, when we finish our a movie or a large lectures, so people go walking outside of the theater, they may want to take off the mask and like the 3D glasses, right, eyeglasses. So they may want to recycle them in a restricted area, so we can reuse it as a resources of plastic. So but the most important thing is that we should pay attention to ourselves, to start with ourselves personally. So pay attention to the do not throw mask unintentionally. That's the most important things. I can see that's the most important, that's the most significant sources of improperly discarded mask on the beach. That's based my personal observation.
I see. Thank you, Dr. He. Yes, waste management is very important. I'm thinking of another equally important issue. It is terrifying me. As you have said in your presentation, we are wearing masks every day. So we are breathing in microplastic every day.
Yeah.
Many hours. I'm thinking if you have any suggestion to reduce the uptake of microplastic from surgery masks, for example, using some new material to replace plastic. As I know, City University of Hong Kong is very good at material science, and there are maybe some other colleague working on some new face mask that can replace the traditional mask.
That's a very good question. Actually, very inspiring. One of our State Key Lab colleague, Dr. Ye, is working on the graphene material to be made on as a key material for the facial mask. The graphene material can kill pathogen, the bacteria and the virus with oxidative stress, with the sunlight as the energy. It's very useful. However, personally, I think when we apply any new material on a mask, particularly contact with the inhalation exposure road, we need to be extremely careful. No one knows whether we breathe in those new material will cause any significant effect or not. And it's so for now on, I believe the facial mask is still necessary for prevention of the diseases. Maybe we can think about wearing fabric fashion mask instead of surgical mask, because those fabric masks are equally useful, and they can they are washable. You can reuse them many times, and they contain less plastic material. So most of them are woven like a natural product instead of the anthropogenic plastic. Likely, but currently we are also working on a follow-up studies to test different facial masks, the N95, the surgical mask, and the fashion mask, to test their potential release of microplastic. For the fashion mask, it's still releasing a certain amount of microplastic, but definitely with less amount compared to surgical mask.
I see. Thank you, Dr. He. Yes, I totally agree. When we think more deeply, perhaps the problem of microplastic from surgical masks may not be that serious. When we consider doctors and nurses in a hospital setting, they wear their masks every day for many years. But it seems that they don't live shorter than us, right? So, perhaps we need more evidence to see the long-term effect of microplastic release from surgery mask.
That's why we first care about the little bug like copepod for the potential receiver of the microplastic that release into the ocean environment. I'm not saying that it's not important to study the human health, but from the current available evidence, I think the microplastic or the nanoplastic may have more significant impact on the ecosystem rather than human health.
Okay. Another angle to look at the problem of microplastic in the atmosphere or from the surgery mask is perhaps when we put on the mask, it prevents us to uptake the particle in the air.
Yeah, sure. Yeah, that's also some potential positive effect, and it has been evidenced that during the COVID-19 pandemic, actually the common influenza, the spread of the common influenza had been significantly reduced. That's proven by a lot of data. So it's we are facing the COVID, but as a result, the common flu has been depressed into a very low level. So when we do this study, we may consider some accidental positive impact, maybe. I'm not sure, but we should certainly look into that potentials.
I see. Thank you, Doctor. Very interesting discussion. It seems that we have gone too far in the air, in the atmosphere. Now we move back to the marine environment. Let's see. I can now see Dr. Zhang Hai is very busy answering questions in the chat box, so I don't disturb him for now. So another speaker, Dr. Marbonok, is not able to check online, but if you have any question to Dr. Marbonok about the regional collaboration, you can type him a message. He is here watching us, okay, and watching the chat box. I have a question for his representative, Dr. Derek Ho. Derek Ho, are you here?
Yeah.
Yeah. In the talk of Dr. Marbolog, he is promoting a regional collaboration of microplastic monitoring. How can we join this collaboration? How can we join the network? I know you're one of the organizers in this regional network.
So previously, if some we outreach to some collaborators and see if they are interested and they can provide some samples to us. So I'll talk more about this, the steps of preparing those samples and how to work on those samples and send to us for the analysis and identification of polymer. But if you want to join this marine bivalves monitoring, it's pretty easy. So collect some samples from a local region, and we'll provide a protocol for the digestion or an extraction of the microplastics from the bivalves, and then you can simply send us the sieve, and then we can help you to identify the microplastics and the polymer type, and also the size and the other related morphometrics for the microplastics. So previously, we have received some samples from Japan and also from Korea from our collaborator already. So we have been working on those data and also those sieve as well. So pretty simple. Just send us the sample and collect from your local S3 or any samples of your interest.
Okay, thank you, Derek. In this collaboration, we have been collecting oyster from different countries in the East Asia region. In the coming years, we will continue working on oyster as well as mussel. Okay, as well as mussel. So we collect both oyster and mussels depending on which species that you have. We have good collaboration with, as Derek said, with Korea, Japan, Taiwan, mainland China. Last year, we have extended to the Southeast Asia region. We have made agreement and collaboration with some university there. After talking with some university professor, we have found out we may need an agreement for some university or for some organization in some country to work out the collaboration. But this is not a problem. We have provided an agreement for university in Philippines and in some other country. I believe it can be done for other countries too. We have another issue. We have another concern report by the collaborator. Sometimes it is not easy to send frozen fresh oyster or mussel, because when we send fresh oyster, it has to be frozen during all the ways of transport. Therefore, we also accept dry oyster or dry mussel. If sending fresh sample can be a concern, you can dry your sample, then send us the dry sample, which is easier for some of you, I believe. Yeah.
And also to add on that is that previously it's very difficult for us to send mussels and oyster from other countries to Hong Kong, and also us sending samples elsewhere because of the COVID. So we have developed a protocol for either digesting the oyster mussel and then send us and filter the samples, and then send us the stainless steel sieve, or send us the dry sample. So in this afternoon, I'll talk more about how to do the microplastics extraction. And even though some of us might not have the filtration kit or the stainless steel sieve, I'll show you how to work on this microplastics extraction using very simple device. So it should be easy for everyone to follow and conduct study in your own area.
I see. Thank you, Derek. So according to the conversation, there are three options, right? The first option is the most direct way to send us the fresh frozen sample, option 1. Option 2 is sending us the dry sample if sending fresh sample is complicated. And option 3 is doing the extraction in your own country, then send out the filter paper with microplastic. In this option, Derek will send you the filtration kit and all required materials from you to carry out the simple extraction, the protocol, of course. Okay, so sounds great, sounds great. Yeah, what else we have not yet mentioned, Derek? By the way, one of the collaborator, Professor Youji Wang from China is here too. He will give another talk about toxicology of microplastic in the afternoon. Okay. Any question from the audience? If not, may I please ask my last question to Dr. Zhang Kai. Dr. Zhang, are you here?
Yes, I'm here.
Yeah, thank you for your presentation. I read some study published a few years ago. They said the removal efficiency of microplastic in some wastewater treatment plants were quite low at that time. But in your presentation, it seems that the efficiency of microplastic removal is quite high. Is it just in Hong Kong or is it worldwide?
Yeah, we have the global average data. It's not very high. If we consider the global average, the removal efficiency for the primary sewage treatment work is only 61%. But in our study, it can be as high as 81%.
Okay, I see. Is the system used in Hong Kong different from other systems?
Yes, the primary sewage treatment work, there are many types of primary sewage treatment work, but for Hong Kong, we choose the chemical enhanced primary treatment work. So maybe this type of primary sewage treatment work have a higher removal efficiency.
Okay. Do you have any data about the particle size that the treatment plants can remove? How small particle they can remove from the wastewater treatment plant?
Actually, from our study, we can see that the most abundant particle size we recovered from the sewage is smaller sized microplastics. So that may imply that the larger size microplastic can be easily removed by the sewage treatment works. But for the smaller sized, maybe it cannot, the removal rate can be lower than the larger sized microplastics.
Okay, I see. Thank you for your sharing. Very interesting. Dear the session chair, we have started this panel's discussion section 5 minutes before the schedule. We may end this section slightly earlier. For your consideration, please.
Yeah, sure. Thank you, Dr. James Fang. So I think here comes to the end of the whole morning session, and much appreciate all the speakers for their nice talk, and thank you all the audience for your kind participation. So see you after a lunch break, and then our afternoon session will start it at 2:10, right? Yes. And the afternoon session chair will be Dr. Yan Meng. So now, thank you all again, and bye-bye. See you this afternoon.
Yeah, thank you, everyone. We will see you in the afternoon at 2:10.
Good afternoon, everyone. Welcome back to East Asia Regional Training Workshop on monitoring and assessment of microplastics in marine environments. My name is Meng Yan, am a scientific officer from State Key Laboratory of Marine Pollution, City University of Hong Kong. And today I'm very happy to serve as the chairperson of the afternoon session. In the afternoon session, we have 4 invited speakers before the free discussion part. And the first invited speaker is Dr. Reg Ho. Dr. Ho is a postdoctoral fellow from Department of Applied Biology and Chemical Technology, the Hong Kong Polytechnic University. So let's welcome Dr. Ho to give us the first presentation.
Hello, can you hear me okay?
Yes, okay, thank you.
Okay, hello everyone. My name is Derek. I'm a postdoctoral fellow at the PolyU now. Today I'm going to talk about the extraction of microplastics from marine biological samples and their polymer identification using Raman spectroscopy. So microplastics has been identified in many environments, such as marine and also terrestrial environments, because of their durability and also the low cost in production. Plastic pollution has been increasingly becoming serious in our natural nature. So because of their small size, microplastics are defined as plastics fragments smaller than 5 mm. So the smallest size of the microplastics will be 1 micron. So for plastics particles smaller than 1 micron, we'll call them as submicron plastics. The abundance of microplastics particles smaller than 300 microns have seldomly been quantified in the marine biological samples because of the difficulties in quantifying the polymer nature and the characteristic span of these plastic particles. Coastal region is one of the hotspots of microplastics pollution because of the proximity to human settlement and the high usage of plastics along this coastline. So the objective of identification and quantifying the microplastics in the biological samples will be to identify different polymer types, to identify the different plastic shapes, and also to quantify the particle size of these microplastics particle. So how do we do this microplastics extraction and identification? So here I give you a very brief overview of the workflow. First of all, of course, we will have to collect the sample of interest. For example, if you are interested in mussels or oysters, then you have to go to the field, collect some biological samples, or if you want to work on fish, or even for myself, I'm a marine mammal ecologist. So I've been focusing part of my PhD study on microplastics ingested by dolphins in Hong Kong. So the first step will be to collect your biological sample, and then we'll extract these microplastic particles from the samples that we collected in these biological samples, and then we'll filter them and then quantify the particle using some spectroscopic measurements. So I'll talk about each step of this workflow in this talk. So the first step is to collect your biological samples, whether you want to investigate the microplastic abundance in the mussels or oysters or fish, or even like what I'm interested in the dolphins. So you'll have to decide whether you want to investigate the microplastics content in the whole sample, or whether you only want to investigate a particular organ. For example, some study will only work on the gut in the fish they collect, or if you want to investigate the particle abundance in a particular organ. For example, for the microplastics in dolphin samples, I only investigated the quantity in the stomach content. So you have to decide which part or which organ that is of your interest, or do you want to identify the microplastics abundance in the whole sample. So of course, you have to take some morphometric measurement, such as the length of the, for example, if you are investigating mussels and oyster, you have to measure the length of the shell and also the width of the shell. And also you have to measure the wet weight or the length of the fish gut or how many other samples, for example, how many fish is inside the dolphin's stomach. So you have to take some morphometric measurement prior to your further procedures for the microplastics extraction. So the next step in microplastics extraction is to digest the tissue. Because of the mussels and like the fish guts or the stomach content in the dolphin sample, it's not easy for us to readily identify the polymer type of the microplastics. So we'll have to use some chemical treatment to extract these microplastic particles for us to continue with the characterization. So for tissue digestion, most of the study, a large amount of study, well, uses alkaline digestion. It's a major way to digest biomass. In the study from our lab, one of the review study conducted recently, over 40%, around 46% of the mussel study and 56% of the fish study uses alkaline digestion for the extraction of microplastics. The use of alkaline digestion is an effective way for removing the biomass, and it has a very mild damage to the plastic integrity. So this part is very important because if you use some strong acid or even sometimes weak acids for biomass digestion, it could cause some surface disruption to the microplastics particle. So most of the study actually uses a very weak, relatively weak alkaline digestion. For example, using 10% potassium hydroxide. So it will be a better extraction method for the identification of microplastics particle. But very often, this study using alkaline digestion will require a subsequent density separation because of certain biomass that are not readily digestible with the alkaline digestion. So around 32% of the study will have to use density separation followed after the alkaline digestion step. So in our study 2 years ago, we identified using the extraction efficiency using different kind of mixture of alkaline digestion using potassium hydroxide together with hydrogen peroxide and also the EDTA. As you can see that we have 3 different treatments with 10% potassium hydroxide, and then in the second treatment we have an extra 30% hydrogen peroxide. And the last treatment, we have 14% EDTA. These 3 combinations were used to test which of the chemical combination will achieve a better digestion efficiencies. And we tested this on 2 different subjects. So the first one is the mussel, and then the second one is the fish biomass. So for this study, we test the 3 treatment, 3 chemical combination under the same condition. So using a 40 degrees Celsius temperature water bath and 48 hours incubation in the water bath. So as you can see that the digestion efficiency is better with the potassium hydroxide, hydrogen peroxide, and EDTA combination, as you can see on the right side of the stainless steel sieve. So this method has been published in the paper 2 years ago. So you can also take a look at the paper and check out the other part of the study as well. So we'll recommend using the potassium hydroxide, hydrogen peroxide plus the EDTA treatment for the chemical digestion for extraction of microplastics. So after so this here I'll give you a workflow for what we do. What we did is that we have the chemical combination here, and then digestion for 48 hours at 40 degrees Celsius. So to give you an example, let's say I have collected some mussel samples or some oyster sample, and then I will measure the width and the shell length, and then I'll also measure the wet weight, and then I'll put them in a Durang bottle. So it's about I'll add about 180 ml of this chemical plus another 20 ml of hydrogen peroxide for the extraction process, which will last for 48 hours. So on a side note, if you want to work on some environmental sample, you can use hydrogen peroxide for digestion, and also for 48 hours at 40 degrees Celsius water bath. So this will let you know the difference between biological samples and also the environmental samples. So the next step of the microplastics extraction after the chemical digestion will be to filter them onto some filtrate so that we can identify the particles on them. So these are the three major type of filters that you will normally see in papers. The first will be very commonly found in laboratory, the glass fiber filter, usually with a very small pore size. So it will be a good filters for the extraction and filtration of a small size particle. And other choice commonly used will be stainless steel mesh. So and then the other one will be the cellulose ester, which is also commonly found in used for chemical filtration. So in order to find the best filters for us to identify the polymer type of the microplastics particle using Raman spectroscopy, we conducted a study using three different size classes of polystyrene beads. And also we measured the Raman intensity of these three filters. So as you can see on the top left, this one is the glass fiber filter. And then the green one will be cellulose ester. And then the blue one is the stainless steel filter. So as you can see that there's almost no peak, no spectrum peak using the stainless steel filter. However, there is a strong signal for the cellulose ester and the glass fiber filter. So this will have an effect on for us to identify the spectral peak for the polymer type of the microplastic particle. So with this, we tested on 3 different sizes of polystyrene beads. We spiked the 10 micron, and then 100 micron, and then 300 micron polystyrene beads on these 3 kind of filter. And then we use the Raman spectroscopy to measure the intensity and see if there is any effect on the spectrum of the polystyrene bead. And we find that the effect is not as severe for the 300 and 100 micron polystyrene beads because of their larger size in the microplastic particle. However, for smaller size microplastics, such as in the 10 micron polystyrene beads, the effect will be stronger for the glass fiber filter and the cellulose ester filter, which sometimes will block the spectrum of the polystyrene spectrum. So as you can see in the bottom three spectrum, the one on the lower end is the 10 micron polystyrene beads. As you can see that the spectrum, the Raman intensity of the glass fiber is actually interfering with the polystyrene characteristic band, which is roughly around at around 950 cm, around at the frequency band at the for this Raman shift. So it also, despite that, the peak of the glass fiber filter is at around 1,300 per cm. It still have an effect on the matching index of the Raman intensity of the polystyrene bead. And the same goes to the cellulose ester. So these two filters, will have an effect on for us to identify the microplastics polymer type. And we found that using a stainless steel mesh, it will have the minimal effect on the characteristics band of the polystyrene bead Raman spectra. So this will be a better choice for the filters that we use to filter the digested material. So how do we do the sample filtration? So we usually use this kind of stainless steel filtration funnel. I can also show you now that is that something like this. So okay, so it's something like this. So we cut the stainless steel mesh in a very small size, roughly around 1 cm. And then we put the stainless steel mesh onto this here. And then we use this funnel to filter out the materials that we have used chemicals to digest on. And also we have these two funnel here, which are 3D printed at the Polytechnic U. So these are the funnel, as you can see, and we make it with different diameter, different size. So okay, so here. So so this will help us to, because of the 3D printing, we can decide whatever size we want to suit our sample filtration. But some of you might ask, so if I don't have this kind of stainless steel funnel, or what can I do? So we, as mentioned by James, we have also, already designed this kind of PVC tube. And if you want to work on the regional assessment of microplastics in bivalves with us, we'll send you one of these kind of filtering funnel. So this filtration kit will consist of two parts. So one is a filtration funnel and a cap. But inside, we can actually put a stainless steel mesh inside. So just like this, this is a 45 mm diameter stainless steel mesh. You can just put it inside. So it will stay on the middle part of this filtration funnel. And then as simple as that, it will create a very simple filtration funnel. And then you can pour your digested material into it, and then use a beaker to collect whatever is filtered out down below. And then you can have your filtered material on this stainless steel mesh. So after that, you can store them in a glass petri dish or in a petri dish, and then you can send us this stainless steel mesh, and we can help you do the Raman identification of the microplastics polymer. So it's very handy and very useful, very easy to handle, even for undergraduates or high school students. So I'm sure that this should be easy to purchase in your own places as well. So you can go check this out and then create some filtration funnel on your own. So for the filtration step, some of you guys may use this kind of glassware filtration set as well. So we also use this for chemical filtration. So for the preparation of the chemical, potassium hydroxide, hydrogen peroxide, and the EDTA, sometimes there are some plastic materials actually occur inside the chemicals because of the purity is not perfect or this chemical grade is not perfect. So we'll first filter out any materials in these chemicals first, and then use the filtered chemical for the microplastics extraction. So we'll first filter the chemical to minimize the microplastics contamination from the chemicals. So some notes for you to be aware of is to wear a lab coat during any laboratory procedure to avoid any contamination. You should always wash your glassware, and then you should also filter the chemicals for the digestion. So you can use as simple as this PVC tube for filtration. So should be easy for everyone to have a try on with this setup. So the last part of the microplastics extraction and identification is the microplastics characterization. So there are many papers that rely on visual screening, which account for roughly 20% in mussels and 15% in fish samples. However, you cannot identify the polymer type of the microplastics particle using visual screening. But this will be a very good way to engage any high school student or university undergrad student to conduct hands-on experimentation. For the identification of microplastics, the most used method is using FTIR followed by Raman spectroscopy. So around 14% in mussels and 8% in fish. So FTIR and Raman spectroscopy are two very similar techniques. However, dealing with different chemical structure, characteristics of the infrared light or the monochromatic light effect. So for FTIR, we measure the infrared range that are absorbed by the material, and then we use the spectrum to identify the polymer type. For Raman spectroscopy, we emit a monochromatic light onto the sample, and we measure the energy loss of the scattered photons. And then we use this for the identification of polymer type. So comparing between the two, both of them are very rapid identification method with non-destructive identification. However, for FTIR, the interference from biological matrix is very limited, but it is very strong. It can have very strong interference from biological matrices using the Raman spectroscopy. For the size of the microplastics particle, there are some technical issue in the lower size detection using FTIR because of the fact that we have to pick the particle onto the FTIR for the identification. So the lowest size that you can detect basically is limited by how small you can pick onto the machine. But there are some improvement with other machinery or the spectroscopies using FTIR, which allows identification of smaller size particles. And also is relatively low sensitivity to impurities in microplastics. So for Raman spectroscopy, we can detect the size of microplastics particle as small as 1 micron. It will require a background spectrum. That's why we have tested that using this stainless steel mesh is a better option than glass fiber filter and also the cellulose acetate. However, the Raman spectroscopy will be having a higher sensitivity to impurities in microplastics. So here are some of the example of the spectrum of the microplastics type that we identified in our samples. So here shows some of the images of the microplastics particle, including PE, PP, PS, PVC, and PET. This is a fiber here, and these are the Raman spectrum of the 5 polymer type here. So using the peak intensity in certain frequency range, we can use this to help us identify what kind of polymer the particle is. And this will help us, we will generate a matrix of a 2-dimensional matrix of matching index, and then we can identify whether this spectrum is corresponding to a certain polymer type. So let me show you a short video here of how the Raman microscope actually works. So this is the Renishaw Raman microscope that we have in the Hong Kong Polytechnic U. So usually if we finish filtering a sample onto a stainless steel mesh, we'll put it onto a glass slide and store inside a glass petri dish to avoid any contamination. And then here we'll put the glass slide onto the microscope stage. And with this Raman spectroscopy, this Renishaw Raman microscope, it can help us generate the whole image of all the particles on the stainless steel mesh. So this is actually one of the samples that I previously prepared for my dolphin stomach sample. As you can see that the image is being scanned by combining many small images on the stainless steel mesh. So this image is important for us because after we generate the Raman spectrum for all the particles on the stainless steel mesh, we can use this to superimpose onto our sample and identify the color and the shape of the microplastic particle. So another thing to note there is that because we use this Raman spectroscopy using a mapping technique, we have not done any subsampling. So all the particles filtered on the mesh, stainless steel mesh is being identified. So there's no subsampling for all our samples. So as you can see that the image, the photo of the whole stainless steel mesh is created here, and then we define the area of interest, and then we set some parameters into for the Raman measurement. And with this, we can generate the whole map of Raman's spectrum on the whole stainless steel mesh. So here shows an example of the finished mapping result, and this is a 2-dimensional array of the all the spectrum that we acquired, and we can generate a 2D image of all these microplastic size and the shape. Okay. So to summarize, this is from what I've done for my stomach sample in the dolphin stomach. So I extract the stomach content, I digest the materials using the potassium hydroxide, EDTA, and the hydrogen peroxide for 48 hours at 40 degrees Celsius water bath. And then I filter them onto a stainless steel filter membrane. And then I conduct the Raman spectroscopy with automated mapping. So because there is a lot of particle on this in this sample, so I have conducted filtration using two different size pore size filter. So the first one is the 250 micron pore size stainless steel mesh, and the other one is the 31 micron. So as you can see, the particle identification shows here is that the particle size is of course a bit larger in the larger pore size filter, and we can also identify smaller particles in the 31 micron filter. So the first step will be to identify the polymer using the Raman spectroscopy, and then we output with a color-coded 2D image, and then we superimpose the image onto the stereo microscope image for the identification of the color and the microplastic shape. So to provide you with some more example is that or some happy news to share is that in PolyU we have recently acquired this optical photothermal IR spectroscopy, which focus the IR beam into the sample surface, which can cause the sample to heat up a bit and create a photothermal response. This machine can allow us to identify microplastics down to a submicron level. So the smallest microplastics that we so far have identified is around 400 nanometer. So it will give us a high spatial resolution with the focus spot size. And also we'll have a new FTIR imaging system at Hong Kong Polytechnic U as well, with a high quality spectrum production with very small micron resolution as well, and with 3 different IR range, which help us to map and identify the microplastics particles faster with a high quality. So with that, I thank you for listening, and I hope if you have any question regarding the microplastics extraction and or the whole laboratory procedure, please feel free to ask me. So thank you.
Okay, thank you, Dr. Ho, for the nice talk and all the demonstrations. And due to the time reason, I think if you have questions to Dr. Ho, you may communicate in the free discussion part later. So let's move on to the next speaker. Thank you, Dr. Ho. Okay, next speaker is Dr. Xu Xiaopeng. Mr. Xu is a PhD candidate from Stakey Laboratory of Marine Pollution and the Department of Chemistry, City University of Hong Kong. His topic today is quantification of microplastics released from vehicle tires in street, dusk, and ambient environment using PYGCMS. Please, Mr. Xu, thank you.
Can you guys see my screen and hear me?
Yes, good, thank you.
Okay, thank you, Dr. Yan, for your introduction. And this is Xu Xiaopeng. My supervisor is Professor Kenneth Liang. And today my topic is the quantification of microplastics released from vehicle tires in street dust and ambient air using pyrolysis GC mass. The tire products are made of the rubber polymer, have been widely used in, for example, vehicle tires, airplane tires, and some waste tires are tied around the pier as the bumper when the ship's passing by. And some other waste tires are shredded into the smaller particles and remake and applied in the artificial turf. And what are the tire wear and road particles? They're very tiny particles released during the vehicle's driving and operation. And tire wear road particles mainly composed of the rubber, which include the styrene rotating rubber, SPR rotating rubber, and the isoprene rubber. And also to improve their physical performance, some tire additives are added during their tires manufacturing. And tire wear and road particles, not just made of the tire wear particle materials itself. It's because of the friction between the raw material and the tire material, and they are the TRWPs are the combination of both. And in next slides, I will just simply call them tire particles instead. And tire particles have been recognized as one of the majority of microplastic until recent years. In China, the model has estimated that about 54% of primary microplastic and 33% of the plastics entering the water body is tire particles. And from this picture, it tells you that in past 10 years, we have a 9% average increasing rate on tire consumption. And in the future, people will also create more tire particles and release into the environment, because we will have more electronic cars, and the electronic cars is heavier than the regular vehicles. And the second picture shows you that the previous research study on the microplastics, you can tell that the PP and PS, PE are most studied types of microplastic. However, tire particles have been ignored as the important source of microplastics, and their environmental data is still leaking now. Okay, the tire particles are mainly produced on roads, and they exist in road dust, and they can be suspended into the atmosphere. And when we on our way to home, to the campus, we are exposed to the microplastics and tire particles all the time. But do we know anything about the human exposure to the microplastics? In previous research, there are many studies have investigated the human exposure of the microplastic in indoor dust, indoor and outdoor dust and suspended particles in the air. However, those research doesn't include the tire particles. Tire particles has been ignored until recent years. But do we want to know anything about the human exposure of the tire particles? Whether they pose a threat to the human, here's the story. And unexplained the death of the coho salmon, which lives in the freshwater system in North America, were found dead since 1970s, especially during the stormwater events. And recently, it was identified the very toxic chemical 6-PBDQ caused their death. What is 6-PBDQ? 6-PBDQ is transformation products from the tire, one of the tire additives, the antioxidant 6-PBD. And 6-PBDQ can even kill the coho salmon, lives in the freshwater system, even at environmental concentration. And their LC50 is about 0.75 micrograms per liter. So I think it is necessary to evaluate the human exposure to the tire particles, because beside of these antioxidant 6-PBD and their transformation products, the tire particles also contains other tire additives like plasticizers, gas linkers, and vulcanization agents and UV stabilizer, as long as we exposed to the tire particles, we are exposed to the tire additives at the same time. So in my research, my question is how much of microplastic, including tire particles, are exposed to the human? My objectives is first to characterize the microplastic in the street dust and the ambient air, which is where is the major source of tire particles, and using the pyrolysis GC mass, which I will introduce it later to you guys. And then to evaluate human exposure via ingestion and inhalation. Okay, for now, I have sampled the environmental street dust and air suspended particles at 2 sites, and they are all in Hong Kong. And at each site, the street dust replicates street dust and air suspended particles were collected. The street dust is collected using a brush made of the pig body hair, and to collect the dust in 30 centimeters times 30 centimeters square. And the air suspended particles is collected using the air sampler here. You can see from this picture that this air sampler is portable, and it allows you to sample the particles above 10 micrometers and from 1 to 10 micrometers. And this air sampling was placed at ground level about 1.5 meters, so that will represent the human respiration level. And after I have 2 samples, and they are treated at a series of the street dust were treated a series of digestion, size separation. They were separated to 6 size groups, and after filtration, they were I would have the dust sample on the glass fiber filter, waiting for afterwards analysis. And the air suspended particles can be analyzed straightly without any other pretreatment. It's pretty easy. And how do we quantify the microplastics mass in my samples? The basic principle is when you have the polymer in your sample and at a very high temperature, about 600 degrees to 700 degrees, they can be thermal decomposed to many smaller molecules. Take the SBR, one of the major rubber polymer as the example. At 600 degrees, they can be decomposed to monomers, dimers, and trimers. At here, I select the dimer, vinyl cyclohexene, as the marker to represent the SBR. As long as I detect the vinyl cyclohexene in my environmental samples, I can know that this is something from SBR. And for other polymers, which is also made to make the tire products, I use also the vinyl cyclohexene and the dipentene to represent the BR polymer and IR polymer. And after I know the mass of the polymer, so I can recover the mass of the tire wear and roll particles, use the fixed ratio. And this ratio is created by the International Standard Organization. So this 1/6.5 is the average ratio between the mass of the polymer and to the mass of the tire wear and roll particles. Then why would I choose the dimer to represent those polymers instead of the tire additives? Because the specificity is very good. As long as you detect these two dimers after the pyrolysis, you know that is something from these 3 polymers. And this polymer is very persistent and not leachable into the environment. And compared to the tire additives, they have the high responses. Here's the small introduction for the pyrolyzer. The pyrolysis GC mass is a combination between a pyrolyzer and a GC mass. And this pyrolyzer allows you to decompose the polymer in a solid sample. That means you can inject the solid sample in the pyrolyzer, and with the helium flow and at a very high temperature, the polymer will be decomposed to many monomers, dimers, and trimers. And then these smaller molecules will be separated in the column and analyzed by the mass afterwards. And in my case, I will set up the pyrolysis mode as the double shot modes. The first mode, I would have the temperature from 100 to 300 to remove the impurities and to remove impurities in the environmental sample, and then finally pyrolyzed at 600 degrees. Other than the tire rubber polymers SBR, BR, and IR, I also analyzed 7 other polymers, which is the commonly seen microplastic in the environment. And here's their MS information, different retention time, the target mass to charge ratio to quantify them, and some other ionics, the mass overcharge ions are selected to make sure that this is the marker we selected. And here is the total ionic strength of I used the standard to inject into the pyrolysis GC mass. And at different time, okay, this is the marker for the SBR and one of the rubber polymer, and this is the IR. And this is basically how we determine the mass of the marker, and then recover to the mass of the polymer. And for the tire particles, and then I use the ratio of the polymer mass over the tire particles mass to recover eventually. Here is some of my results. This is the microplastic concentration in street dust at site 1 and site 2. At site 1, the PP has the highest proportional, and followed behind by the tire wear and roll particles as the brown colors. And the highest at the site 2 in the street dust, the tire particles has the highest proportional in the street dust. And the average of the microplastic concentration in street dust is about 3,000 micrograms per grams. We can see that the PE and the PE has the highest concentration, and then followed by the tire particles. And next, here is the results of microplastic concentration in the air suspended particles in PM10. And in this figure, it's different from the microplastic in the street dust. The tire particles has the highest concentration, and followed behind by the PP. PE here. And the average And the average concentration in the air suspended particles is about 2 micrograms per cubic meters. After I have the microplastic concentration in the street dust and the air suspended particles, I calculated their human exposure via the ingestion. Here I assume that the children would ingest 200 micrograms per day if they're exposed to the street dust, and adult as the 100 micrograms per day. And outdoor workers are exposed to higher street dust, 330 micrograms per liter. And at site 1, the concentration of tire particles is okay, not very high than the PE, but the site 2 is the ingestion of the tire particles almost higher as the all other microplastics. Okay, that's all. Actually, I haven't analyzed all my samples, and this is just a result of 2 sampling sites. And I'm also looking forward to share my research outcomes in the future about the human exposure via ingestion and inhalation. Thank you for listening.
Thank you, Mr. Xu. Thank you for the nice presentation. And we may have 5 minutes for Q&A. Any questions from the audience?
Hello.
Hi, hello.
Yeah, may I please ask a question?
Yeah, please.
Yeah, thank you, Shopping. Very interesting talk, very important. You have suggested another way, which is very powerful to analyze microplastic. I have a question. In your method, you use some degradation product, for example, some dimer as a marker to indicate microplastic particle. Okay. Because I'm not familiar with the method, I wonder how confident we are to say this dimer or other detected compound come from the particle, the microplastic, but not from other volatile compounds.
Because these particles are the human-made polymers, and we have the few blanks. We have the few blanks to remove. Let's just say if you paralyze these sediments first time to decompose all the plastic polymers, and then if you decompose the second time, there will be no signal of these polymers at all, of these markers at all.
I see. Very interesting.
Hi, Mr. Xu. There's one question we can see from the chat box. Hi, thank you for your presentation. Can you be a little more specific about the codification of TWRPS using GC mass? I guess you may need standards of those dimers.
No, I don't actually have these standard of dimers. I have the standards of the polymers. So yeah, if I know the mass of the polymers, I ingest it into the equipment, and then I have the response of these dimers, and then I would be able to make a curve between the relationship of the mass of the polymer and the responses of the dimers. Hope that will answer your questions. Okay.
Thank you. We have time for more questions. You are always encouraged to use the chat box.
Okay, could you explain sampling duration for the ambient air?
Did you ask a question?
Yeah, let me know the sampling duration for the ambient air.
The what? Sorry.
Sampling duration time.
Here, did you mean the microplastics in air particles?
Yeah.
Okay, what do you want to know?
Could you explain the time duration for the sampling of microplastic particle in ambient air?
Did you say time duration? Okay. Okay.
Yeah. Yeah.
Thank you. Here. So this sampler kept sampling the air for 48 hours with the flow 20 liters per minute. And so the volume of the air would be about 28 cubic meters.
Okay, thank you.
Thank you. I noticed Dr. Xu Xiaoyu raise your hands, right? You can ask question.
Yeah, I just pressed the wrong button, but it's okay. Thanks for Ms. Xu's sharing. And actually, do you have some you mentioned you mentioned have samples from road dust and from the atmosphere. Do you have more sample, like other environmental samples?
Yeah, I have the river sediment sample, marine sediment sample, but I had a problem that the type particles may exist in there, but if I just ingested the sediment samples directly with the simple treatment, for example, like digestion or digestion using the Fenton reagent, but still the background is still too high. So I'm not really sure whether type particles in there or not. But other polymers like the PP and PE can be detected instead of the type particles.
Thank you. Thank you, Mr. Xu. Thank you for presentation. And you may notice there are some other questions from the chat box. Please respond to the questions. By using the chat box, we need move on to the next speaker. So please end your share screen.
Okay.
Thank you. Okay, let me introduce the next invited speaker, Professor You-Ji Wang. Professor Wang is from College of Fisheries and Life Science, Shanghai Ocean University. And the topic today is ecotoxicological effects of micro and nanoplastics and multiple environmental stressors. Welcome, Professor Wang.
Good afternoon, everyone. I'm You-Ji Wang, come from Shanghai Ocean University. Also, I'm an alumni of City University of Hong Kong, where I got my PhD degree in 2011. I studied with Dr. S.G. Zhang and Paul Xin in the Department of Biology and Chemistry. Thank you very much for Professor Kenneth Liang's invitation for this workshop. Today it's my great honor to present my microplastic research in the past few years. The title is Ecotoxicological Effects of Nano and Microplastics under Multiple Environmental Stressors. We focus on developing a unique marine animal health evaluation system in response to multiple environmental stressors, which has guiding and application significance for marine agriculture and marine ecological evaluation. The environmental stressors you can see include hypoxia, warming, ocean acidification, metals, microplastics, and so on. And the fitness measures include physiology, metabolism, immunology, behavior, growth, and survival. So the upper right corner is the official WeChat QR code of our group. And the lower right corner is the QR code of Marine Environmental Research, a journal, which I serve as an associate editor. You are welcome to scan and subscribe to see the latest research progress of our group and the journal. So microplastics are plastic fibers, films, particles with diameter less than 5 millimeters. The annual production of plastics worldwide is more than 300 million tons every year since 2014, and the production is increasing every year. Microplastics come from a wide range of sources and enter the marine environment by various routes. Finally, they can accumulate in various marine animals as well as human bodies through consuming seafood. In this talk, I will introduce 3 parts of microplastic research. The first is field investigation and risk assessment. The second is toxicity testing of microplastics in mussels and fish. And the third is about the combined effects of microplastics with other environmental factors on mussels, and also a case about crab. In the past few years, we mostly have investigated some typical sea bays around East China Sea. For example, the Hangzhou Bay and the Xiangshan Bay, where we studied the microplastic pollution in surface seawater, sediments, marine organisms using some recognized sampling and analytical method. Then we also conducted some routine lab treatments, analysis, and identification of microplastics for in the morning, and also just Derek and Shopping have talked a lot about the methods aspects. As I know, James' team has achieved a good progress in this area. So I will not talk about this contents on the analytical methods. The Hangzhou Bay, you can see, is economically developed and densely populated. The interaction between human activities and marine environment is apparent. Complicated hydrodynamic conditions and the fast-growing economic development have put the microplastic pollution in Hangzhou Bay waters into increasingly severe situation. So from this figure, we divided the Hangzhou Bay into 3 parts according to geospatial development and utilization. This is South Bay, mainly is for industry, urban use, and marine culture and fishery zones. The central region and the north region contain some marine culture and fishery zones. Then in the past few years, we measured the microplastic pollution in surface water, sediment, and biota of these sites. So the following 6 polymer types were identified in the water, sediment, and biota samples. You can see cellulose, PE, PET, PP, and PE, and rayon. The surface water samples contained fewer polymer types than the sediments and biota samples, being dominated by PE and PP, with lower amount of rayon and cellulose, especially PE and PP, which have a high global demand, were more abundant in South Bay samples. So PE type, PE microplastic is dominant in biota, and rayon is dominant in sediments. The abundance of microplastic can be found in fish, shrimp, and crabs. The type and quantity of microplastic from fish is slightly higher than that in shrimp and crabs. Microplastics in organisms may be attributed to the aging fragment from agricultural products such as fumes, fishing nets, and floating boats. For microplastic abundance in surface water and sediments of Hangzhou Bay, the microplastic abundance at South Bay are significantly higher than in the central region and North Bay. Residential life and industrial manufacture may be the dominant sources of microplastic in the marine environment. So the influence of geospatial development and the utilization of coastal areas may influence the distribution of the microplastics. So you can see from this table, the average abundance of microplastics in surface water of Hangzhou Bay actually is relatively lower than other coastal waters in China. The results of microplastic pollution in Hangzhou Bay possess a certain value for reference. Nonetheless, corresponding data standardization and analysis remain to be improved. So the Xiangshan Bay is a typical agriculture zone that has been in operation for many years, and a larger number of typical commercial agriculture species, such as fish, shrimp, enter the fish market from there. In 2016, the agriculture activity at Xiangshan Bay were investigated on site to verify the interpretation of remote sensing information. Satellite remote sensing analysis shows that agriculture activity at Xiangshan Bay in 2016 were mainly composed of oyster farming, kelp culture, and fish cage culture, which occupied a large area of this bay. For the distribution of microplastic in sediments, the abundance of microplastic in Xiangshan sediments generally decreased from the inner bay. This is the inner bay. From the inner bay to the bay mouth. Agricultural activities such as oyster farming may elevate microplastic pollution in sediments. Another reason is the weak hydrodynamics in the inner bay reinforced the accumulation of microplastics. The inner bay is more vulnerable to human activities, especially discharge from domestic sewage. So the size of microplastics ranged from 300 to 5,000 micrometer, with an average of nearly 2,000 micrometer. Over 70% of the microplastics range from 500 micrometer to 2,500 micrometer, while only 7% range from 3,500 to 5,000 micrometer. You can see the fibers gradually increased from inner bay to the bay mouth. The bay mouth is about 80%, and also intensive agriculture activities in the inner bay probably generate a huge amount of fibers from aging nets and ropes. In terms of the number of items per soft tissue weight, both fish species had lower microplastic abundance than the other three shellfish species. The lower microplastic abundance in fish is due to our observation that microplastics were found only in the intestine and stomach, but not muscle. The muscle accounts for 40 to 60 of the total biomass. In terms of the number of items per individual, we can see there is no significant difference among the species. The size of the microplastics was mostly between 70 and 1,500 micrometer in organism, but this size range was slightly lower than that in sediments. Such difference may be mediated by the biological process. For example, mastication or digestion after ingestion of the microplastics, which could reduce the size of microplastics. So the value of the three indices, for example, H, PLI, and RI for the water in the entire bay was estimated to be 22.7, 1.9, and 128.4 respectively. These three indices shows similar spatial variation patterns, which indicate that the ecological risk in the inner bay was higher than the other two regions. So, than the other two regions. So, according to the criteria for microplastic pollution risk level in water, the overall ecological risk in the water across the entire bay was ranked as low to moderate. The microplastic in the organisms had an average size 1.3 millimeter with diameters less than 2 millimeter particles, accounting for more than 80% of the total particles. The trophic level of organisms range from 2 to 4. The relative microplastic abundance was negatively correlated with the trophic level. The trophic magnification factor of the microplastic in the organism was 0.1. Therefore, microplastic did not show trophic transfer or bioaccumulation. in organism. So the second part is about the toxicity testing of microplastic. We first test the ingestion of microplastic in muscles. Microplastics with 5 particle size were exposed to muscles. The accumulation of particles in gill, digestive tract, and mantle of muscles were measured after 87 hours exposure and also following a depuration period. The intake rate of microplastic from water by muscles were efficient and rapid. Microplastic intake is size dependent. You can see small particle size are more likely to be ingested by muscles. The accumulation of microplastics was highest in digestive tract, but microplastic in digestive tract decreased significantly after depuration period. We found unstable accumulation of microplastic in gills of muscles. Microplastics in digestive tract increase with the exposure time. Also, we think microplastic may reach the mantle by means of in-body transfer. We expose muscle to 0, 10, 10,000 and 1 million item per liter microplastic concentrations. And also we recovered the muscle for 7 days after 14 days exposure. The effects of microplastics on biogenetics of muscles were assessed by cellular energy allocation approach. We also reviewed the toxicity mechanism of PS microplastics in marine muscles by high coverage quantitative metabolomics using chemical isotope labeling liquid chromatography. From this figure, we can see carbohydrate is consumed first, followed by fat and protein. Carbohydrate is the earliest nutrient to replenish recovered significantly after the recovery period. As an important substance, protein is also consumed, indicating a serious lack of energy. So available energy significantly decreased, and also the energy consumption significantly increased under the high microplastic concentration. From this figure, we can see medium to high concentration of microplastic can exacerbate the energy expenditure of muscles, which has a long-term negative impacts on energy budget of muscles, and also increase the consumption of their own substance. The cellular energy allocation was significantly reduced and could not be fully recovered within 7 days after exposure to microplastic at medium and high concentration of microplastics. So during the microplastic exposure, the increase of ROS were found by the increase of lipid peroxidation index, for example, MDA and LPO. This significant upward trends were only observed in 10,000 and 1 billion particle per liter microplastic groups compared with the control group. And there was no significant difference between 10 particle per liter and the control group. Similarly, previous study have shown that microplastic can cause significant effects on oxidation state of bivalve, including changes in antioxidant enzyme. However, during the recovery period, this is a recovery period. During the recovery period, this significant difference disappeared, suggesting causal association between bioaccumulation and oxidative stress. As for the immune parameters, we can see the medium and high particle concentration significantly increase the ACP and ALP activities of muscles, and they also increase significantly with the exposure time. So the exposure of microplastics significantly increase the activity of ACP and ALP, suggesting immune response of muscle under the pressure of microplastic. They can resist the toxicity of microplastics. Similar to the trend of oxidative stress indicators, during the recovery period, we can see the ACP and ALP activities returned to the same level as the control group. So this results further implies a causal relationship between microplastic accumulation and the measured effects. We also measure the mRNA expression of heat shock protein SHP70, which was significantly upregulated after 14 days exposure to the high concentration. And it also increased with the increase of exposure time. So heat shock proteins play a vital role in protecting cells from damage and repairing damaged proteins. The toxicity of microplastics mainly involved metabolism pathways of some amino acid, for example, histidine, vitamin B6, tyrosine, phenylalanine, and beta-alanine. During the recovery period, all differential metabolites levels did not differ from those before exposure. This is a metabolic pathway of muscle exposed to microplastics. We can see that there are mainly 5 amino acids involved in TCA circle. The TCA circle is the center of the interconnected circle of the energy transmission. Changes in this metabolic pathway can interfere with TCA circle and affect energy supply of the cell. And it further affects immunity, metabolism, and growth. The high correlation between immune parameters and the changing trends of differential metabolites generate our hypothesis. Microplastic can cause significant change in metabolism of 5 amino acids in muscle hemolymph, affecting antioxidant and immune parameters. Moreover, significant difference in metabolite levels were also observed at environmentally relevant concentrations. For example, 10 and 10,000 particles per liter of microplastics. However, during the recovery, most indicators recovered. So we turn to how about the microplastic accumulation through food chain and the relevant toxic effects on high trophic level organisms, such as crab. This chain is about microplastic algae to mussel to crab. So can a biomagnification of microplastics happen in this food chain? And how about the body defense of the crab? We measured some parameters. You can see the tissue accumulation of microplastics in crab. The order from high to low is hepatopancreas, intestine, gill, and muscle. The accumulation of microplastics in the combined exposure routes was more than that in the waterborne and the foodborne alone. Microplastic, we can see, can induce toxicity and oxidative stress, decrease the activity of detoxifying enzyme erode and antioxidant enzyme GST, and also nerve damage. When the concentration of accumulated microplastics reach a certain level, the level of ALT and AST increased immediately, resulting in liver injury. So muscle have a higher capacity to accumulate microplastics, which means higher BCF, because muscle have a better filtration capacity than crabs. Microplastic concentration in crabs was less than that in mussels, and even in the combined exposure group, the BMF was less than 1. This means microplastic did not biomagnify. So as microplastic pollution is expected to worsen in the future, microplastics may disrupt the adaptability and viability of marine organisms, thereby affecting the stability of marine ecosystem. So now we move to the effects of nanoplastics on fish. This is a case of a commercial fish, large yellow croaker. From this experiment, after 14 days exposure, in the high nanoplastic concentration group, the activities of antioxidant enzymes in the liver of the fish increased, leading to oxidative stress. After 7 days recovery, the activity of antioxidant enzyme in the liver of fish in high concentration group was still higher, but did not cause lipid peroxidation. So juvenile fish to try to maintain the stability of intestinal environment by increasing the number of intestinal probiotics, but this cannot eliminate the adverse effects on the digestive tract and the immune dysfunction. Also, the collapse of the self-regulation ability of the fish. So these results indicate that there are three bacterial phyla in intestine of fish, of juvenile fish. The growth of large yellow croaker is closely related to the proteobacteria and Formocasts, which could be more effective for getting energy from the diets. And the lactobacillus affiliated to the Formocast is generally considered to be closely related to the health of fish and has the function of preventing and treating disease of the digestive system. This is about microbiome. We can find high mortality in the microplastic groups. We think nanoplastic can cross the self-regulation of large yellow croaker. Also, we found the lower specific growth rate in experimental groups. We think nanoplastic limit the nutrient uptake efficiency of fish because we can see various digestive enzymes decreased. From this figure, we can summarize lysozyme, trypsin, and lipase of fish were reduced under nanoplastic exposure. Growth and survival of fish were reduced under nanoplastic exposure. Dominant bacterial phyla in the gunfish changed significantly under the nanoplastic exposure. A significant increase in potentially pathogenic bacteria was observed under nanoplastic exposure. So the third part is about the combined effects of microplastics with other environmental factors on mussels, and also there is a case for crab. So this figure is made by Genbo by Else, the next speaker. Apart from plastic waste, our ocean is also impacted by ocean acidification. Since the ocean acidification and microplastic can trigger adverse biological effects, so their co-occurrence would pose a great challenge to the survival and health of marine organisms. In this experiment, we expose mussel to full concentration of microplastics under 2 pH levels. One is acidification. And we measure the digestive enzyme and antioxidant enzyme. We found high concentration of microplastics, and acidification inhibited the activity of all digestive enzymes. Acidification can reinforce the negative effects of microplastic. Even during the recovery period, some digestive enzyme can recover, but trypsin and lipase were still inhibited at high concentration of microplastic and acidification. So we think acidification can enhance the negative effects of microplastics on some parameters. For example, GSH. GSH, and also it can be but it can be recovered in the recovery period. The other two SOD GPX activities were not affected by acidification and microplastic exposure. This is about the antioxidant activities. So in this study, the mussel were exposed to microplastic and acidification. Both stressors had more severe effects on digestive enzyme than the antioxidant enzymes, although the negative effects on certain digestive enzymes shows a reversible trend. More biochemical indicators have not recovered. For the physiological aspects, under the normal pH, respiration rate of mussel were lowered by microplastics, but acidification caused a reduction in respiration rate. At normal pH levels, we can see clearance rate was unaffected by microplastic at low concentration, but greatly decreased at high concentration of microplastic. Acidification negatively reduced the clearance rate. Assimilation efficiency was lowered by microplastics, whereas a further reduction was driven by ocean acidification. Absorption rate was reduced by acidification, regardless of microplastic concentration. At normal pH, we can see the extraction rate was lowered by microplastics. However, reduction in excretion rate by microplastic already occurred at a medium microplastic concentration. By integrating the physiological parameters, we can see the energy budget decreased with increasing microplastic concentration, and also was reduced by ocean acidification. So we found acidification and microplastic reduce hemocyte concentration and viability, possibly due to elevated oxidative stress mediated by ROS production. Acidification and microplastic can adversely affect the immune system, limiting the phagocytic energy and reducing the hemocyte viability by inducing oxidative stress. Also, we measure the biases. Biases are important for mussel survival. muscle-based production is hammered by acidification and microplastics, which are associated with reduced energy budget. In addition to biases abundance, declining biases quality due to acidification and microplastic may further increase the risk of the muscle survival. So in conclusion, for this study, ocean acidification reduced phagocytic activity and hence immunity of muscles. The reduced phagocytic activity was associated with lowered energy budget. Acidification also reduced biaxial performance. Microplastic can aggravate these negative effects of ocean acidification. Also, muscle would be more prone to disease and dislodgement in future oceans. So our findings suggest that co-occurrence of acidification and microplastic pollution would increase the susceptibility of muscles to infectious disease, thereby threatening their survival and undermining their ecological contributions to marine community. So microplastic can also interact with organic pollutants when they mixed with organic pollutants, and microplastic can act as a carrier to become a rich collective of organic pollutants. The effects of microplastics on muscle physiology after absorption, environmental pollutants need to be clarified. So in order to explore the toxic effects of BDE, BDE is an organic pollutant combined with microplastics on marine muscles. We expose the muscles to microplastics in combination with BDE for 3 weeks to determine the immune defense, oxidative stress, and energy metabolism. We can find the feeding rate decreased after microplastic or BDE explorer alone. BDE inhibited the oxygen consumption of muscles. Microplastic exposure alone did not affect oxygen consumption, but the combination of these two significantly increased the oxygen consumption. BDE exposure had little effects on the lactate dehydrogenase activity, but the combination of these two decreased its activity. So microplastics significantly increased the ACP and ALP activities. Low concentration of BDE did not induce the LPO and MDA. So ROS and MDA were significantly increased after the combined exposure. We can see microplastic and high concentration of BDE inhibited the heat shock response in muscles. So in summary, muscle reduced their food intake, tried to adjust physiological activities by increasing respiration and changing the V energy supplied under the combined stress of BDE and microplastics. Although no mortality was found during the experiment, we think muscle living under this pressure for a longer time would face inflammatory reactions and finally lower growth and survival. So the last case is about the Chinese mutton crab. So this is a very popular food in Shanghai. Microplastic may be potential vectors for environmental contaminants such as heavy metal in water due to their highly hydrophilic surfaces. to investigate the combined effects of microplastic and the lead. We expose Chinese mud crab to different microplastic and lead concentration for 3 weeks to determine the lead bioaccumulation, oxidative stress, lipid anabolites, and histopathology of Hepatopancreas. In general, the results showed that compared to single lead exposure, the combination of microplastic and lead significantly increased the bioaccumulation of lead and activities of antioxidant biomarkers and lipid metabolism, and also the liver injury parameters in crabs. So our results indicate microplastic are potential vector of heavy metals, and co-exposure exerts more severe effects on crabs. So this study provides insights into the oxidative defense and preliminary lipid metabolism of economic shellfish in response to combined stress of heavy metal and microplastics. So in conclusion, we have carried out a field investigation and analysis of microplastics in natural water bodies, and we also conducted some methodology explorations. On this basis, we have carried out toxic effects of microplastics on various animals, including fish, mussel, crab, and also we measured some physiological activities, for example, ingestion, food chain transmission, and also immune and physiological activities. In addition, we simulated the complexity of the real marine environment and carry out the compound ecotoxicological effects of microplastics under multiple environmental stressors. So in future, we should carry out in-depth health risk assessment of microplastic from ecological and global scales. So this is a relevant publication of this talk, and this is an acknowledgement for our microplastic research in mainland China, and also James, also the next speaker, Iris, and also collaborators from Germany. And also, I would like to thank my students. A lot of students have worked very hard on this topic. So this is the end of my presentation. Thank you very much.
Thank you, Professor Wang. Thank you for your nice talk. It's very informative. And due to the time reason, we need to have a break, 10 minutes break. And if you have questions to Professor Wang, you can use the chat box. You can communicate with Professor Wang during the open discussion session later. Thank you. We may come back 10 minutes later. Thank you. Hello, everyone. Welcome back. Next speaker is Dr. Elvis Xu. Dr. Xu is Associate Professor from Department of Biology, University of Southern Denmark. And the presentation topic is about toxicity of micro and nanoplastics across taxonomic groups. Welcome, Dr. Xu. Thank you.
Thank you, Chair. Just want to make sure if you guys can see my cover slide.
Yes, okay.
And can hear me? Okay.
Yes, okay.
Perfect. Very glad and happy to share my I'll say most recent 5 years works about toxicity of micro and nanoplastics. So in this talk, I would like to share my reflections and a lot of questions actually during the past 5 years working on this topic. Very tough topic to work on to an environmental toxicologist. And thank you very much for attending this. And thanks for PEMSEA and Kenny for the invitation. So I think not all the audience are experts, or they are experts in the audience, but in this presentation, I would like to give a more general discussion-like presentation. So first, please allow me to give acknowledgement to my, you can see a lot of global collaborators, including Yoji, who is the previous speaker. And I'm an associate professor and a spare research leader founded by Denmark DFF. And I'm from the ecotoxicological group from University of Southern Denmark. And this is what we do in our ecotox group. And basically, we do basic and applied ecotoxicology research focus on endocrine disrupting chemicals, heavy metals, and more recently, I would say 5 years, plastic particle research. This is what we are. And a little bit back to plastic facts. First off, I think we should be fair that not blame plastic. Plastic is a great materials. The first fully synthetic polymers actually is made not 5 years ago, not 50 years ago, but in fact, it's 115 years ago. We already start to have the ability to make polymer as plastic materials. However, because it's just so good, it's just overcome a lot of things that we can never do before we have this material. So plastic has been used in almost all aspects, all sections of our society. And after 100 years, then this becomes a problem. This has become a big top threat, arguably the top threats to our globe. Not only the ocean, but the microplastic starting from the ocean issues. So here you can say, arguably, we have oil spills, we have ocean dumping, we have land runoff, waste from ships, dredgings, and plastic waste is now attract more and more attentions to researchers, to the public, and to government. Just please look at the numbers here. And the plastic waste, we're not talk about the plastic production, but the plastic waste into the aquatic system, it's 6 million tons per year. And if we do a simple calculation, it's equal to every minute we'll dump a full truck of plastic into the ocean. And according to a most recent OECD report, this number will be doubled, and in some cases will be tripled by 2060. So 2060 is not that far away, and all of us probably will see this, but nobody wants to see this. And because of that projection, then a lot of scientists start to move their efforts and focus to try to win this combat with plastic pollution. So this on the left side, you can see this is a very recent Nature review paper that called to prioritize this plastic pollution. So it is a global issue. But why microplastic is, as I said very earlier, why is it so difficult to study? So first of all, this is so little and so tiny, and you already heard from Yoji's study, the resources is so diverse, and there is no boundary. Why there's no boundary, I would like to share with you these two recent publications on science advances and one in Nature Communication. So what the findings in common from these both studies that indicate that there's no boundary. So both of the studies go to the most remote far environment to sample either seawater or either sample from the snow or ice. Guess what? No surprise. Or it's surprisingly when it published that they always find microplastic. So these two studies indicate again that there's just no boundary of microplastic contamination. And there's another story that I would like to share. Actually, if you look at this beautiful photo there, that is a photo I took 10 years ago in a beautiful marine reserve in Hong Kong. But below that, you can see it's the same place, it's the same area, it's the same year that we find a lot of trash that is found in this protected, which is a no-take area. And a lot of them, or most of them actually, are plastic waste. And after 10 years, we have now enough evidence, enough monitoring data can have the more up-to-date status of what is marine protected areas can be protected from microplastic. The sad news is that no, it cannot, because if you look at our more recent publication, environmental pollution, is that we can find enough evidence to show 2/3 of the available data reporting that microplastic level in the global marine protected area was higher than 12,000 items per square kilometer. So this is indeed a global question, a global issue. Then the next question is, where do those microplastics come from? And as Yoji also said, it's very diverse, but actually the major contributor of those environmental microplastics is highly depending on the regions. For example, this one is from the Norwegian Environmental Agency, and the number one is actually from the tear and tear from the car tails. And then the number two is the synthetic text clothing, and then the loss from the plastic products. The list is long, but it's just example that is the major contributor of this environmental microplastic is depending on where. And because of that report, collaboration with Chinese professors, we did a lot of sampling, try to figure out where those environmental microplastics come from. And before this study, of course, there's already a lot of nice environmental monitoring study. But in this study, we more try to answer this question or to test this hypothesis that whether the sediment microplastic are somehow related to the soil macro and microplastic. So to do that, we designed to sample soil and sediment that is physically very close to each other. And the hypothesis is that if the distance is so close to each other, then the microplastic profile in these two environmental matrices should be similar. But unexpectedly that we did not see this, and we don't find any obvious relationship between the microplastic profile, even they are sampled in adjacent soil and sediment. So the one possible or plausible explanation to this is because the sources are too complex that we cannot simply identify their sources. And over time, the sources can also be varied, and I'm talking about the number one sources could also be switched if we have new sources like the mask. And unfortunately, I didn't attend the talk by Harry this morning, but actually we also have this concern like almost two years ago about these new sources. So the single-use face masks, and 99% of the disposable surgical mask actually is made of plastic, different layers of plastic. If you think the plastic bulk waste in the environment is a big source of the secondary microplastic, then the face mask is a more bigger problem because it is made directly from the micro, even nano-sized plastic fibers. And then, of course, in the water, then over time it could degrade into microplastic. It has been proved by Harry's study. And however, we are still doing some studies to try to quantify the release of micro and even nano particles from the mask. And the study is still ongoing, and we do find some different findings to Henry's paper, and I would like to share it once it's published. So it's just example that the number one contributor to original environmental microplastic could switch if we have a new bigger source of microplastic. And so far, if you notice, I haven't talked much about nanoplastic. And simple definitions very similar to microplastic is depending on the size, right? Although the size range is is not raised consensus, but most of the research agree that if we have a plastic items that is smaller than one micrometer, we call it nanoplastic. And it's called the lost 99% in the ocean. Why is that? Because based on the historical data from the first day that we start to use and produce plastic materials to date, we can kind of estimate how many plastic have been produced. And we also have the global monitoring data on the ocean. We can also modeling and estimate how many microplastic are already in the ocean. But when you compare the two data and the plastic that we already know in the environment to only account to 1% of the total plastic that have been produced. So the question is, what is the lost 99%? And of course, there could be a lot of reasons, but one hypothesis that a lot of labs are testing is probably over the 100 years, 50 years, the microplastic in the nature environment already degrade and fragment into the smallest plastic particles. We call it nanoplastic. And this is what my research focused on in the past 5 years. And of course, it's not only my lab, and a lot of more labs are interested in this topic. And this is just a quick Google search and compare the number of nanoplastic in the title in Nature journals and all journals. You can obviously see a sharp increasing research focus on this topic. And here I just would like to highlight one of the perspective paper. I think as an environmental toxicologist, this is highly recommend to read this paper. It's called Nanoplastic Are Neither Microplastic nor Engineered Nanoparticles. And I just read one sentence why nanoplastic are not engineered nanoparticles because of this high particle heterogeneity originate from various sources, environmental conditions that result in nanoscale materials with a wide range of sizes, shapes, and overall composition. And even the nanoplastic originate from the common source. And from a toxicological perspective, it's also because when you have a particle that is so small compared to the micro, then it has higher potential to pass the biological membranes either from passive diffusion or can even be actively transported intercellularly. So this is a nice perspective paper, give us a lot of reflection to rethink about whether we should seriously take nanoplastic as different new emerging contaminants. So that's why my past 5 years really most of my time are committed to answer nanoplastic related questions. And again, I would like to give acknowledgement to a lot of global collaborators to generate a lot of new and useful information and try to reach to some more general conclusion. And we do find something in common, and we do find something not in common, and we cannot answer all of the questions. As you see here, we do find that doesn't matter what species or marine or terrestrial or even bacteria or algae, they always can be directly exposed to our life, and our humans are no exception to that. That has been approved and shown by not our study, but a lot of studies. And another thing is just so widely distributed almost in all ecosystem. If you still remember the two studies I shared is in the Arctic environment. And fortunately, there's no acute effects. And okay, you can argue that if you dose them like a gram per kilogram or gram per liter, we do see some effect. However, in general, if you talk about more environmental relevant exposure scenario, then we don't see any acute effects. But unfortunately, we always see some sublethal effects, and it's due to not the particle itself, but a mixture of nanoplastic, microplastic, and on top of everything is a leaching chemicals that could be more toxic than the particle itself. the mode of actions, the exact mode of action, unfortunately, we don't have a precise answer yet. And another bigger question is the quantity. So far, it's still very hard or challenging to quantify the nanoplastic in the environment or in biological samples and environmental samples. So this is what I have in common. But in today's presentation, I will give you three case studies to dive a little bit deeper into some of the study we published previously. The first one is I want to talk about the chronic effects, but only look at the most simple model, polystyrene nanobeads as a model, primary nanoplastic as an example, and in the simplest aquatic animals as the toxicological model. And next, I would talk a little bit about the chronic effects of the more real-life plastic leachate that contains secondary micro and nanoplastic. And the list will talk about the common toxicological effects and the potential mechanisms across different taxonomic groups. All right, so this is the first study. Before that, we still need to know that we are not there yet to understand the toxicity of plastic in the environment, especially the nanoplastic. Why is that? So as an environmental toxicologist, reviewing a lot of papers and read a lot of papers, we find that most of the toxicological study on nanoplastic use well-defined single polymer types, Bs, and we know the mass concentration. However, in reality, in the nature environment, the complexity level is much higher than we thought. It has different polymer types, different morphology, surface characteristic, different particle sizes, and we don't know the particle concentration yet, and the chemical absorption and desorption is another hot topic. So all of those point that we still need a lot of study to understand the toxic effect in a real life environmental condition. So this is why we do this. So we, as I said, because of so complex nanoplastic in the environment is not nanoplastic itself. So I think when we talk about plastic toxic effects, either micro or nano, we should specify what type of plastic you are talking about or you are assessing here. So here is the polystyrene nanobeads, use a model primary nanoplastic. And in this study, we choose the simplest organism, Daphnia, as a model. As you see, it's transparent, it's relatively big, and it's easy to culture and short life stage. So just use this simple organisms, we still find a lot of problems that we thought we are expert as an ecotoxic or environmental toxicologist, but we don't have actually experience to study nanoplastic in the first place. I tell you why. So as an environmental toxicologist, we always try to quantify the toxicity based on their dose response effect, right? And this is what we find. It's beautiful. We can calculate the LC50, we can calculate the EC50 of nanoplastic and microplastic when we use the commercially available polystyrene nanoplastic or microplastic beads. So problem solved. We can just do as we did before for PAH, for PCBs, for different endocrine disrupting chemicals or heavy metals. But is that the truth? So we have a question mark whether this is a true toxicity. Can the nanoplastic beads, beautiful actually, the beads transparent, can be that toxic? So when we repeat the plastic beads after we thoroughly wash the plastic beads, this is what happened. We find no effect at all. Doesn't matter what size of the particles we tested, it just cannot be repeated, this result. Why is that? So we try to figure out what caused this high toxicity of this nano plastic and microplastic. Then we look back to the providers and we find that it is actually the sodium iodide that is used in the suspension that provided commercially. So this is what caused the nanoplastic toxicity. It's not the plastic or micro or nanoplastic bead itself. So we are learning from this. So and we published the study Environmental Science and Technology Letters a couple of years ago. But surprisingly, to date, I still see a lot of researchers actually still using the same source of commercially available bees without take care of the washing or treatment of their bees to do toxicological tests and get some conclusions. So if we know that it's not from the particle itself and the particle itself is not that toxic, then we try to lower down the concentration and we try to extend the exposure time, see whether we see some adverse effects of those two bees. So the next experiment, we try to do multi-generational exposure, again, using this simple animal model. And this is experimental design. We only expose the parents and we do not expose the F1, F2 generations. We want to see whether this effect is absorbed or not, whether this adverse effects can be transferred to the future generation. And this is photos that we show that even the for F1 and F2 generation are not exposed to nanoplastic directly, but they can get the nanoplastic from their parents. So it's a direct evidence that the nanoplastic can be detected in future generation, even after generations of recovery. So it can be, then the next question is, okay, if you notice that's very bright green, and we also find some background autofluorescence signals, then the next question is, okay, again, we don't do this when we are assessing the toxicity of other chemical compounds, but for nanoplastic, if you want to use fluorescence label, then you need to be more careful about whether the fluorescence is indicator of nanoplastic or it's just a dye. Right. So to answer this question, we do some Leachy experiment. Basically, we use a dialysis concept. If the particles are, if the dye can be leached out from the nanoplastic itself, then they can make this silicon as a acceptor green. And after this leaching experiment, if you look at the photos on the left, indeed these dye are not that stable. They can be easily leached from the commercially provided nanoplastic and then light up the silicon, which means that they are not stable. The green fluorescence you see probably is not 100% a nanoplastic bead. It can also be the dye itself. So it's a very big problem if you try to use those fluorescence nanoplastic beads to look at, for example, the cellular uptake or the organ or tissue distribution of nanoplastic, and you need to be very careful. And then what is the solution? Then using dialysis concept, then we can dialyze the fluorescence labeled nanoplastic very thoroughly. And this is the experiment we did. So after the dialysis for 1 week and 2 weeks, you can see that the nanoplastic fluorescence labeled, the fluorescence are more stable. Then we can use the cleaned up nanoplastic to toxicological study. And then the question is whether, okay, if you did dialysis, then maybe you also change the behavior of nanoplastic in your solution. So to test that, we do this analysis before and after dialysis, and we find that dialysis itself are not changing these diameter, for example, of the aggregation of nanoplastic. So we are safe. We can do this dialysis, and this is more suitable to do a cellular uptake experiment, for example. And after we clean the bees and we do the toxicological test, then this is what we find after 2 generations of exposure. And it's very different to if you don't do the dialysis. And you can say it's just they can decrease the heart rate, it can decrease the reproduction, and can affect the uptake and decrease the some movement. And overall, it has not acute toxicity test. toxicity effects. So the message from this study is that the nanoplastic, polystyrene nanoplastic beads exhibit limited but transgenerational effects in Daphnia. And washing nanoplastic beads is necessary, especially if you use fluorescence labeled commercially available nanoplastic beads. That is message number one. And remember, that is the commercially available model nano polystyrene beads we use. But in reality, the situation is more complex than polystyrene nano beads. So we want to see the chronic effects of leachate from the weathered plastic waste, for example, a very complex mixture of different type of micro and nanoplastic, and assumably also plastic compounds that could be released from in your during your exposure. Again, we use Daphnia as a model. This time, the experiment design is simple. We just go to the supermarket, we buy random nanoplastic products, and we cut in pieces. We put in clean water, pure water, and clean glass flask, and we put on the roof on the UV for 20 days, and we use a leachate dilute to different concentration to test whether they have acute effects and chronic effects. Right. So after 20 days, this is what we find. We find millions of nano and micro beads, particles in the plastic leachate. Means that under the UV light, it's not difficult to get secondary nanoplastic and microplastic in your exposure water. And we found a lot of them attached to the surface of the Daphnia. Probably some of them can already be uptake by the Daphnia during the exposure. Remember, it's a long-term exposure. And if you look at the distribution, it can be very small, and we can confirm that many of them are plastic of different polymer types. So more nanoplastic than microplastic. And also, not only the plastic particles can be released from those plastic waste, but also a lot of organic and inorganic contaminants that could be released from into the leachate. And a lot of chemicals are unknown, but there are some chemicals we are very familiar with. For example, the bisphenol A, due very toxic and high endocrine disrupting effects. And also we have a lot of heavy metals detected in the plastic leachate. If we know that there has a lot of toxic chemicals in the leachate and a lot of heavy metals in the leachate, then it's not surprising to see that the plastic leachate can adversely affect Daphnia. So I will not go to all the details. You can say it's different to when you expose Daphnia to the single polymer type polystyrene nano beads. So it's just, if you compare these two studies, you should have this question the same as me, that when we are talking about the toxicity of nanoplastic, we should specify which type of polymer types in what size, whether this is really reflect the mixture situation in the environment. So message number 2, plastic leachate show no acute effects, but the observed sublethal effects were due to the mixture of secondary nanoplastic, microplastic, and leaching chemicals. That is my message too. And then the last study, as a case study I want to share, is this the publication we published 2 years ago about the key mechanisms of micro and nanoplastic toxicity cross taxonomic groups. So as I briefly mentioned, we have actually some years of study on this topic about microplastic and about nanoplastic. And in fact, if you Google it, we published 7,000 papers last year about micro and nanoplastic. So we have enough knowledge, at least with the in vitro assay, to predict what are the potential toxicity toxicity or key mechanisms across different taxonomic groups. So this is the study we meta-analyzed around 100 papers. This is a conclusion we get. In general, the toxicity mechanisms of this MMP, depending on their size, surface characteristics. Surface characteristics sometimes has more effect than the size itself, but we have relative understanding, limited understanding of the charges, and not all the toxicological study consider the surface charges, for example, and polymer types as well as the cell types. And it's makes sense because different cell types respond differently to nanoplastic exposure. And the plausible toxicity mechanism mainly include the membrane disruption, extracellular polymetric substance disruption, ROS generation, DNA damages. So you can read the details about our findings, but what we find in common is summarized here. Okay, and if you say I'm a marine biologist or I care more about human health, then the figure A could be something you are more interested in. So in animal cells, the positively charged nanoplastic can, for example, indicated in yellow, smaller. If it's smaller than 500 nanometer, then it has a potential to destabilize the lysosome and initiative a cascade of cellular damages. And if you say that I'm more interested in the plant and say the crops or other algae, then you look at the number, the figure B. So the MMP can also affect the growth of plants by indirectly altering the soil properties. And if you say that I are really interested in the algae, then you look at the figure C. So in the plant cell, nanoplastic can be taken up through the disrupted and damaged membranes, aquaporin and cell pores, and further cause ROS generation and damage to the photosynthetic system. And if you say that, so what about the bacteria? Then you look at the figure D. Then for bacteria, the MMP can be trapped by the extracellular polymer layers, the EPS, and where the MMP damaged EPS can result in a decrease of hydrolysis activity and surface charges and increase of ROS. So ROS appears a lot in different studies across different taxonomy groups. So we suspect that this is a major toxicity mechanisms in common. Then the last, this probably is one of my last slide. So what about human? What about human health? Do we have evidence to show it's also bad to humans? Then I will tell you that we do have a lot of in vitro evidence to show that probably is not good thing that you eat a lot of nanoplastic, microplastic. That's why we put together a lot of available information we put into this special issue in Environmental International called Nano Microplastic Toxicity and Human Health and Health Effects. And more recently, we also, well, I think it's one month earlier, we published this new study called Nanoplastic. Is it a new cardiovascular risk factor? And in Environment International again. And here you can see there's a lot of hypotheses about the exposure rules, about how it can get into the circular system and what are the possible mechanisms of cardiovascular toxicity and what are the potential symptoms or pharmaceutical endpoint that is expected. And of course, all of the, not all, but most of the study are based on either human cell lines or mice and mouse models or fish models. And so far, although there's many increasing knowledge about the potential health effects, adverse health effects to humans, we still don't have a direct effect to indicate whether nanoplastic is doing bad things to our own body or how. All right, so with that, I think I would end my talk today, and any question is welcome. Thank you.
Thank you, Dr. Xu. It's an excellent talk. And there are two questions from the chat box. I think we can put the Q&A session into the open discussion session to be led by Dr. Fong, our chairman. So Dr. Fong, you may lead the discussion part. Thank you.
Yeah, sure. Thank you, the section chair, and thank you all the speakers who have provided very excellent talk. We have received some questions in the chat box. Let me some of them for Professor Wang. Professor Wang. The first question is, Wilma is asking if there are environmental factors to be measured during sampling for microplastic research, for example, for the muscle research. What are the important factors that need to be measured during the sampling, please? Professor Wang.
Okay. So I don't know if you want to learn about the field research or the lab research, the laboratory research. If you want to know the sampling, microplastic sampling in field research, I need to see that during the sampling, we mostly measure some other environmental factors such as dissolved oxygen, salinity, chlorophyll, and also some other factors we think that they may influence the microplastic distribution in the field. For example, we recently just finished a work on the microplastic pollution in Xiangshan, in Hangzhou Bay. We measured the salinity. We found that salinity will influence the microplastic distribution in the seawater. So if possible, for the field study, you can measure as many as more environmental factors during your research, during your microplastic research. And if you want to, for the exposure experiment in the lab, I think the most important factor you need to measure, I think, is about the cultural conditions for the animals. For example, temperature, salinity, pH, dissolved oxygen, ammonia, like this. And also, if you add additional environmental factors such as some chemicals, organic or inorganic pollutants such as copper, lead, or other some metals, you need to measure the concentration of the pollutant in the seawater. Also, for the effects on the organism, you also need to measure this pollutants accumulated in the animal body. I think this is a factors during the sampling for field research or for the lab exposure research. Okay.
Yeah, thank you, Professor Wang. You are beloved by the audience. There are 3 more questions for you here. The first question is, how often do we use aquaculture plastic? As you know, there are a lot of plastic used in aquaculture. How often we use the plastic aquaculture for we do marine research? The second question is, are you aware of any report that report the toxicity of human due to consumption of microplastic from fish product? Okay. Question number 3 is about the toxicity of polyethylene. He said in a city, they use polyethylene to do the water distribution pipe. Do you think if there's any concern about the use of polyethylene? Thank you.
Okay, okay. So for the, I think for the second question, I think Elvis, just the last slide of his lecture just shows the toxic effects on human health. And so because we can expose to microplastic in the air, in the food, and also in some drinks, and also we need to breathe. Sometimes some plastic particles in the air we can ingest in our, for example, in our lung. And for seafood consumption, I think if the particle is nanoparticles, is nano microplastics, then we may have a chance to absorb in our body through a circulatory system. Because previously, I think maybe a few months ago, I just see a paper that nanoplastic was found in human blood. And so I think, although we did not heard hazardous effects of micro or nanoplastic on human health, but I think it's not good thing to find nanoparticles in our blood. And also we can see that maybe there are some other routines for nanoplastic exposure. I think maybe later Dr. Genbo Xu may talk about this in more details. So for the first question, for the agriculture activities, because I don't know in other countries, but in China, for agriculture activities in some, for example, in our research in the Xiangshan Bay, a lot of plastic products have been used in such activities, such as some fishing nets. The main components are plastic, and also for the net culture of some fish, we also use some, for example, some floating balls. I see that these balls are made of, I think is a foam, because these foams are very easy to be broken by, I think, no matter by sunshine, by UV, by other physical pressures. So because these materials, these plastic materials are cheap in production, in agriculture. So a lot of fish farmers, they, in order to reduce the cost for agriculture, they use some cheap plastic products. In this way, a lot of plastic, they will broken into small particles. So this will increase the plastic particles in the seawater and also in the sediments. So a good news is that I heard last year, for example, there is the largest mussel agriculture area in China. I mean, in Zhoushan Island, the Shengsi country. So this is the largest mussel agriculture area in China. I heard that the government announced a policy, and they will replace the cheap plastic tools in mussel agriculture, and they will use some products which will be more strong and will not be easily be broken into plastic particles. And they also value the situation of microplastic pollution. So they want to have some updates for the agriculture facilities to replace some cheap microplastic products. Okay, so this is for the agriculture activities. During this process, a lot of microplastic have been used. And the last question is for distribution pipe to household. So using the plastic pipe. I think this is not a serious problem, because normally for the daily water use in Shanghai, we also use plastic tube or pipe, and this kind of pipe, I think, is not easy to be broken, because this pipe normally will be, I think, will be covered by no matter in the wall or under. I mean, they will be wrapped. So I think the pipe is not easy to be broken. So I think it's not very dangerous. Also, maybe a few of particles will be broken for longer time. But I think this is from my point of view. I think now it is safe because human health, I think, is the first place we need to consider. So I think the authority and the government will consider this issue. We don't need to worry about this, and more we need to consider is about other issues, such as the mask, agriculture production, and also some plastic rubbish that are managed by people. Okay, so this is my reply to these questions.
Oh, thank you, Professor Ong, for the detailed answer. We have received two other questions to Dr. Zhu. The first question is if we can analyze nanoplastic and microplastic directly from Daphnia sample without fluorescence. The second question is, what do you think are the key challenges when you work on the exposure experiment in Daphnia?
So the first question is whether we can directly measure. Yeah, so if you talk about quantification of mass, and yes, I think one of the previous talk by Xu says we can use Paratis MS to quantify the mass of micro and nanoplastic. I think that way we can quantify the mass of nanoplastic, but directly measure the nanoplastic particle, I'm talking about particle concentration, is still not possible to my knowledge today. The next question is, that's why we want to add fluorescence as a indirect indicator to show where the particles goes instead of look at the overall mass load in Daphnia. And the next question is the challenge of setting Daphnia exposure experiment. Is that the question?
The second question is the major challenge when you work on Daphnia.
Well, so Daphnia is a, if the question is related to whether Daphnia is a great, is good to use or easy to use, then it's easy answer is yes, it's a great model, it's very easy to culture, easy to use, can use for chronic, transgenerational, multi-generational, long-term exposure, no problem. If you talk about if there's any key challenges of doing nanoplastic exposure, yes, there are a lot of challenges. For example, related to the previous question, you cannot quantify the particle concentration, the tissue concentration yet for nanoplastic. If you want to explain, for example, the toxicodynamics of nanoplastic, today it's still a big challenge. Yeah.
Yeah, thank you, Dr. Zhu. Further to your question, our lab has developed two methods to quantify small particle smaller than 1 micron in our sample. The first method is the use of OPTRL, which is optical photothermal infrared spectrometry. We can analyze particle down to 400 nm. Another method is surface enhanced Raman scattering, the SERS technique, that we can analyze particle around down to half micron. So the technique is new, but we are trying to optimize to make it more easily to be used.
Yeah, that's great method, and I saw two publications. Both are in Nature journals using this technology actually to analyze sub-microplastic particles in food products. I think there are two publications in Nature, so great method. But again, I would like to comment, if I'm allowed, that if you look at the review paper I showed about the potential toxicity mechanisms across taxonomy groups, and the problem is particles smaller than 200 nano. That is problem. So we still have the size limitation of detection or quantification. So, yeah.
Yeah, sure. If that, we have to work harder then.
Exactly. Yeah.
To find more method.
Yeah.
Okay.
Need fundings, need more researchers.
Okay. Any question from the audience?
James, I have a question for all of the speakers. So based on the European report and also other recent reports showing that the vehicle tie microplastics seems a very important microplastics. However, there are not many studies actually working on them. I wonder, first, is it limited by the technology for monitoring? And second, would this tie particle actually degrade over time? Because I heard that some of the components actually similar to electro bubbler. So I want to hear from Xiaopan and maybe Eugene and Elvis to see what you think. Based on Xiaopan's work, it seems to just the dust on road dust, we can find quite a lot of them. They are quite in par with the PE microplastic as well. So I want to listen to your view. Thank you.
Thanks, Kenny. I think it's a very important topic. It's getting hotter and hotter in Europe in general about tire microplastic. So in fact, I published one paper about tire microplastic 3 years ago in PNAS. And from the finding, we think that the particle itself, okay, we have a lot of knowledge about particle toxicology, and seems the particle toxicity of rubber microplastic are not that high. That caused most of the toxicity, at least based on our research, is that it's the leached chemicals. For example, the benzoyl triazole, which is causing a lot of the toxicity from the tire microplastic. So in my opinion is that when we talk about microplastic toxicity in general, we should also consider hundred and thousand of chemical that is leached over time from the particles, in addition to the probably minor particle toxicity itself. Yeah.
I also wonder, because right now the method you use to screen the microplastic, you're using a flotation method. You may miss them because this plastic will sink to the bottom, right?
Yeah, well, we are doing some cool works about the fate of plastic, microplastic in the ocean. For example, we collect sample from waters below ocean, 7,000 meters below the water, and we still found plastic. Definitely, the particles can sink into the deepest ocean. So in the environment, it also find in the environment, the plastic also form a lot of biofumes and colonized by bacteria and other fungi, and they can definitely sink. Yes.
Yeah, Professor Leung, perhaps I provide some more information. You're absolutely correct. In the monitoring paper in the past few years, not many people reporting the Thai microplastic, but in a most recent report by Europe, Thai microplastic contribute a large proportion in the microplastic pool. I think the problem may be the analytical technique that we use to analyze microplastic. You're absolutely right. We cannot use the flotation method because a lot of plastic will sink. Therefore, currently, most of the researchers, they use digestion method. They digest the organic matter or other unwanted material from the particle itself. Another concern is the use of Raman spectroscopy and FTL spectroscopy. They are rather weak on black surface. Rubber are usually black. They absorb a lot of light. Therefore, this particle may not be that easily detected by the conventional method. For this reason, we are going to use the new OPTIO to detect the black rubber materials. Perhaps Dr. Derek Ho can share with us his experience. Is Derek here?
Yeah, so we probably also haven't it's not easy to detect with from but recently we tried out from that with
very cool collection.
Yeah, Derek, the connection is very poor at your side. It's okay, perhaps I say something for you. Yeah, if the collection is bad. We have collected some Thai wearing material from other researchers, and we have started to optimize the method to analyze this type of plastic. It is an ongoing work. So in the future, we expect we may see a higher abundance of Thai microplastic in the environment.
If I can comment to what James said, I totally agree that the current methodology developed for like the traditional plastic polymer doesn't readily applicable to tire microplastic. So called dark plastic cannot be analyzed using FTR or Raman. So we also published a paper about black plastic problems earlier this year. So if you're interested, I can send you the link about the dark or black plastic issues.
Yeah, that would be great. I'd love to read that paper. Is there any more question from the audience? If not, perhaps I ask one more question to Dr. Su. A rather simple question. How do you define nanoplastic? Do you use 1,000 micron or do you use 1 micron or 0.1 micron? How to define nanoplastic?
Yeah, I think this is still, well, in our lab, we define nanoplastic as smaller than 1 micrometer. Yeah, but when you talk to, if you talk to a lot of the bio tech guys, then they don't agree. They say smaller than 100 nano is more suitable to define nano. But I mean, it depends the questions. In my opinion, yeah, there's no physical cutoff, which is nano, which is micro. And the more important is whether we can find any physical cutoff that shows toxic effect that affect the health. That is, to me, is more important.
I see, I agree. We have received one more question from Rudolph Fo in the chat box. What is the best solution for the flotation of microplastic? Flotation is not ideal because when you add the high density solution to flow microplastics, some even higher density microplastic will sink to the bottom, just like the Thai microplastic. As we have just discussed, perhaps we can use another approach, the digestion approach. We try to digest the unwanted materials to get the plastic particle. Yeah. But sometimes it is not easy if you have a sediment sample in which a lot of sand particle that you want to separate. It depends on the situation.
Okay, maybe I have the final question for all of you. How can we reduce microplastics and nanoplastics for the oceans? A billion-dollar question for you.
Yeah, while we are thinking about this question, there's another question asking by Xiaopang. What is the size limit to analyze single plastic particles in ramen? The theoretical limit is 1 micron, but the practical limit is about 2 to 5 micron when you use ramen. To answer the question of Professor Kenneth Leung, perhaps we can make some device to trap microplastic from the source. For example, washing machine. I know you have published a paper to put this type of special device in some laundry machine, right? That can be one of the proposal. I don't know. Perhaps we can upscale it to be put in a sewage treatment plant.
Yeah, I think I'm following up with the global treaty on plastic pollution. I don't know if you also are following that. I think there's a lot of debate and discussion on how to win this plastic pollution modern issue. So it's not my opinion, but it's the discussion point from this global treaty on plastic pollution is that we definitely need a life cycle assessment and prevention. So it's not like closing the tap, not let the water run. It's not simple solution like that. And some countries, well, first of all, it's not the one country can do this. So some country suggest it should bottom up. Some is the opposite. But what in general so far agreed is that we definitely need to do a life cycle assessment from the source to the production, to the transportation, to the waste management, disposal, and on top of everything is the business.
Yeah, I agree. I think now it's about the time. If no more questions from the audience, perhaps I give the time back to the section chair.
Okay.
Thank you.
Thank you. Thank you, Chairman. And next, let me introduce Dr. Yu Wittner and invite Dr. Yu Wittner to give closing remarks. Dr. Yu Wittner is the president of PEMSEA Network of Learning Centers and the director of Center for Coastal and Marine Resources Studies, IPB, University Indonesia. Let us welcome Dr. Yu Wittner to give closing remarks and acknowledgements. Welcome.
Thank you, Yan. You hear my voice?
Yes, yes.
Yeah, thank you. Good afternoon, ladies and gentlemen. The Honorable Executive Director PEMSEA Amy Gonzalez and Professor Kenneth Leung, SKLMP, City University of Hong Kong. As a preliminary of my closing this afternoon, I introduced about my activity last week. We have an attended of the World Ocean Assessment regional workshop for large marine ecosystem in Indian Ocean, Pacific Ocean, Asia, and Australia Ocean. One of the message from this regional forum of the World Ocean Assessment is the concerning for the increasing and participation of all marine countries against marine pollution, including marine plastic. We have just this point, pandemic COVID and also and potential global recession. Often we are also struggling to improving the quality of the marine environment, facing continuously high population growth, health of environment, food quality is a challenge that needed to be realized next. One of these challenges is the macro and microplastic as pollution on the coastal and marine ecosystem. As emerging pollutant from organic and inorganic source down to the sea of the various part of the world. From the previous presentation and the first presentation this morning, and then that speak by James Punks, that microplastic not only in the water, but also in the sediment, including in biota and species. Both of the sample this morning, Dr. Fang explained in the fish and mussels. The information still show that the challenge for the quality food are also work to be done. In responding this challenge, collaboration between university, research institute, and NGOs needs to be strengthened in East Asia. In the training and the workshops, I saw the partner from the university attending also in this workshop, local government also attending ICT, and also NGO and other parties. And my expectation is that SKLMP and University of Hong Kong and also the other PNLC members can continue to develop and prepare the more advanced training, capacity development, and capacity building for the ASEAN countries and the world, and starting for handling, sample technique analysis, interpretation, and also how to define the strategic action for the mitigation and prevention. PEMSEA Network Learning Center with PNLC and also PEMSEA, together with PEMSEA, we design a training mechanism and system to be accommodated in the Asian region throughout the world of this pollution control and monitoring. The strength of the PNLC member is how the many of our activity and program are be able to accepted by coastal and marine communities such as university, local government in East Asia. The indicator is how much they receive advocacy, assistance, including training from us, PNLC member, in the important to the implement. To accept that they're free of methodology, tool, and instrument that we offer in solving problem on the coastal and marine. In this context, our partnership with PNLC in each country is key of the implementation on integrated coastal and marine management. And our program support to government to providing sustainability and welfare of the coastal community. In this occasion, I would like to express my high appreciation to all of speakers who have delivered material from this morning to this afternoon. Professor Kenneth Loong, as the director of SKLMP in City University of Hong Kong, Professor Yeo Ji Wang, colleague of Fisheries and Life Science, Shanghai Ocean University, Dr. Elvis Chin Bozu, Associate Professor, Department of Biology, University of Southern Denmark, Dr. James Carl Hei Fang, Assistant Professor, SKLMP and Hong Kong Polytechnic University, Dr. Henry Yue, SKLMP and City University, of Hong Kong, Dr. Kai Zhang, Macau University of Science and Technology, Macau, Dr. Derek Yuan Wahu, the Hong Kong Polytechnic University, Dr. Elizalde Eshebum Mabolak, the Hong Kong Polytechnic University, and Mr. Shopping Zhu, SKLMP and City University of Hong Kong. Based on the presentation of the speaker, we have many information about source of the plastic, research methodology, and the analysis and also instrument. And that we also found the impact and risk on the ecosystem and human health. At this moment, I would like also thanks to all of the organizing committee, both of SKLMP and the University of Hong Kong, under coordinating Professor Kenneth Sloan, James Fangs, Yang Meng, Mang Wei, Yang Jingshan, and Sora Chung. And also from PEMSEA, Amy, Karen, Ingrid, Nancy, and Thomas. Finally, I also thanks to all the participants that attending in this event. The insight that have become by the speaker can strengthening the capacity of the participant in the monitoring program for plastic debris in the water, sediment, and aquatic biota. Along with this, make this opportunity to strengthen our network and capacity at the local level as well as global community awareness in joint research, joint publication, and working together across countries in East Asia. Don't hesitate to communicate with SKLMP, speaker, and the training partner from various countries, because this is a moment for us to communication between parties. With the completion of this event, I convey the message that the microplastic monitoring workshop and training is complete and officially finished. Thank you very much for the participation and see you in the next training held by PNLC and PEMSEA. Good afternoon. Regard from Bogor, Yon Fitner. Thank you.
Thank you. Thank you.
Thank you, Dr. Yon Fitner. And see Professor Leung and Dr. Fong, see if you want to supplement.
So I like to echo to the president. So I want to thank everyone joining this workshop. Without you, we can't make this a successful training workshop. Today, really happy at the maximum time, we have 132 people participating in this workshop. The number is so great. It's a very good encouragement for us to organizing more this similar type of training for the region. Once again, thank you, Dr. James Fan, for leading this effort.
Thank you all the speaker and all the audience joining us today. We have been talking about this workshop for quite a years. Finally, we have the opportunity. We are very happy to share the research findings, and hopefully in the future, we can establish a more official research collaboration in the regional scale to provide more baseline data on microplastic in East Asia. Thank you.
Thank you.
Thank you.
Thank you, Dr.
Bye
Fong.
bye.
Yeah, bye-bye. And that's the end of the training workshop today. Thank you for your time. Bye-bye.
Bye.
Bye.